to wait a few minutes to make sure that we're synchronized with the online audience. Good morning to all of the participants and attendees of the Regulatory Education for Industry Annual Conference 2024. After the inspiring and informative plenary session, we are ready to move on to each center's track with many exciting talks scheduled ahead of us for these couple of days. It's a great pleasure to welcome everyone to the CBER track, in which speakers from the Center for Biologics Evaluation and Research will be presenting on a variety of scientific and regulatory topics related to investigational and approved biological products. My name is Larissa, and I'm the Associate Director in the Division of Clinical Evaluation in the Office of Therapeutic Products in the Center for Biologics Evaluation and Research. And this is my fourth time meeting with the CBER track attendees. As in previous years, with a number of colleague moderators, I will be hosting the CBER track, which will run today and tomorrow. Now, many of you know that CBER regulates a wide variety of biological products, and they include cellular and gene therapy products, blood products, tissue products, vaccines, allergenics, xenotransplantation products, antivenins, and even some devices used with biological products. And every year, when we plan the conference agenda, we post pre-conference polling questions, and we learn from those questions. Most of them, our potential attendees respond to correctly, but sometimes we see surprising results, as we've seen in one of the questions that I'm going to show you in a minute. And the results do tell us that the REdI conference is needed to continue improving knowledge about CBER and FDA and about what we do and which products we regulate. So here's the poll question sent through LinkedIn. Center for Biologics Evaluation and Research regulates all of the listed below except. And here you have the choices, and you see that the total number of voters was 1,816, and most responders said that we don't regulate devices, whereas the correct question was bispecific antibodies. Bispecific antibodies, as well as monoclonal antibodies, belong to the category of therapeutic biological proteins that are regulated by our sister center, Center for Drugs, whereas devices in general, of course, are regulated by the Center for Devices and Radiological Health. CBER does regulate devices used with some of the biologics, and today, during session 3, in the talk of Cara, she will talk about processes that are used by CBER, which are similar to what's used by the Center for Devices when we process the device applications. Now, as you have heard earlier today, the overarching theme of our educational event this year is innovation. And where else, of course, if not here in CBER, will you hear about innovative approaches to medical product development? And so we will start our track presentations with the session that would be including talks about genome editing technologies. And later in the track, you will also hear about advanced manufacturing and the use of artificial intelligence in the vaccine development and much more. So innovation and progress, of course, cannot occur without a lot of communication. And in CBER, we believe that good communication and engagement of stakeholders, and particularly patient engagement, really is the foundation for successful product development. And so we made a special effort in our today's agenda to include presentations on communications related to specific product development questions, as well as inquiries that come to CBER, particularly on the tissue wall, and of course on the engagement of stakeholders in CBER's work. So while discussing the communications with the Center for Biologics, our speakers will also discuss the best practices for interactions between CBER and product sponsors, because these and many other issues remain relevant and important for smaller companies, particularly at the early stages, at times when they transition from academic research to product development and try to increase and improve their production and manufacturing. And as in previous years, all of our presenters in the CBER track are experienced reviewers or expert policy staff, and they base their presentations on product programs and applications which they previously reviewed or discussed with product developers. Let me now introduce the CBER track conference team, or our MC team, and you will see these folks here in the room today and tomorrow, and our online audience will hear their friendly voices. So our online audience this year is much larger than our in-person audience, and the two people who will be interacting with the online audience, which is a possible task, would be Adrienne Fisher and Ohai Dirizu. Let me just introduce them quickly. They're there in the back of the room. You can come say hello to them during the break. Adrienne is Regulatory Project Manager in the Office of Review Management and Regulatory Review in the Office of Therapeutic Products in CBER. She joined the FDA in September of 2018. It was like yesterday. I still remember interviewing her. And ever since, she has provided review management through the product lifecycle, from pre-submission meetings to the post-marketing submissions. And prior to joining FDA, Adrienne served as a public health analyst within the Office of the National Coordinator for Health Information Technology at the Department of Health and Human Services. Adrienne received her Master's of Public Health, Environmental and Occupational Health degree from Florida International University in Miami, and her MBA from Webster's University in St. Louis. Ohai Dirizu is also a regulatory project manager in the same office in CBER. Both Adrienne and Ohai serve as liaisons between FDA and industry, managing various communications, meetings, and regulatory submissions. Prior to his work at CBER, Ohai served as a consumer safety officer in the Office of Medical Device and Radiological Health Operations. He received his Bachelor's Science degree from Northwest Florida State College and his Master of Science in Regulatory Sciences from Johns Hopkins University. So Adrienne and Ohai will be interacting with the online audience both days. Our moderators for day one of CBER track for today are Graham Price and Cecilia Crowley. Dr. Price is the Chief of Gene Therapy Branch 5 in CBER's Office of Gene Therapy. He received his PhD degree in Biological Sciences from the University of Birmingham, United Kingdom, and then went on to pursue his research interests in the Medical College of Georgia. Dr. Price is, in fact, an FDA veteran with expertise in immunology and virology. He has close to 18 years of experience of working in CBER. In his regulatory role, he reviews INDs and BLAs for a variety of human cell and gene therapy products. His main expertise, though, is in chimeric antigen receptor T-cell products and hematopoietic stem cell-based therapies. Ms. Crowley has been with the agency since 2008, and currently she serves as a regulatory project manager in the Office of Therapeutic Products. She oversees management of diverse portfolio of regulatory submission and provides excellent customer service to our industry stakeholders. So those are our moderators for today, and I'll give the podium to Graham in a minute. For tomorrow, Melek, Sanai, and I will moderate day 2, and I'll just briefly introduce Melek before we dive into our first day topics. So Melek holds a PhD in pathobiology from Johns Hopkins University. She completed her postdoctoral fellowship at the United States Army Medical Research Institute of Infectious Disease in 2020, and right after that joined FDA. Initially worked as a pharmacology toxicology reviewer, and now serves as the team leader in the Division 2 in Office of Pharmacology Toxicology in the Office of Therapeutic Products. So this is our MC team, and I would like to now remind everyone to please silence your cell phones. Let me now give the microphone to Dr. Price, who will start our first session today. Thank you.
Thanks, Larissa. Okay, good morning. I'd like to thank Larissa for that introduction. It's my pleasure to introduce 2 excellent speakers this morning who will be giving you very informative talks, and then that will be followed by a short question and answer session once both presentations are concluded. So first off, we have Brian Stultz, who is the Chief of Gene Therapy Branch 2 in the Office of Gene Therapy CMC within the Office of Therapeutic Products at CBER. Brian has over 19 years experience with cell and gene therapy CMC review, including expertise in plasmids, peptides, mRNA, genome editing, and AAV-based products. He received his Master of Science with training in biochemistry and molecular biology from the University of Virginia, and joined the FDA in 2000, where he worked in the laboratory of Dr. Deborah Hirsch, publishing multiple papers on the regulation of TGF-beta signaling and identifying critical quality attributes of mesenchymal stem cells. In addition to research, he participated reviewing gene therapy and eventually transitioned to full-time review and in the Office of Gene Therapies. He's currently the Chief of Gene Therapy Branch 2. Gopa Raychaudhuri is the Associate Director for Special Programs in the Office of the Director at CBER. She's engaged in facilitating development of gene therapies for rare diseases, including engagement in private-public partnerships, including the Bespoke Gene Therapy Consortium. She serves as liaison to a World Health Organization, providing strategic direction and overseeing CBER's scientific collaborations with WHO. Dr. Raychaudhuri has long-standing interest in the development and access to essential medical products to promote national and global public health. She received her bachelor's and master's of science degrees in biochemistry from the University of Toronto and a PhD in microbiology from the University of Virginia. Her postdoctoral training at NIH and NIAID focused on vaccine development, and since 2010 she has been in her current position where she implements strategic programs to advance CBER priorities, including the development of gene therapies for rare diseases. So now I'll hand it over to Brian for his talk, Regulatory Approaches for Gene Therapies Incorporating Human Somatic Genome Editing. Brian, over to you.
Thank you, Graham, for that kind introduction. And I wanted to welcome everybody here to the CBER track this morning, everybody that's in the room, and of course, the many people that are attending virtually. So as I was introduced, my name is Brian Stultz. I'm a branch chief in the Office of Gene Therapy CMC, and the topic of my talk today is going to be regulatory approaches for gene therapies incorporating human somatic genome editing. Okay, covered that. So I've divided my talk into 2 main sections. The first, I'm going to cover the basic genome editing technologies, how they work, what they are, hopefully give you a basic understanding. And the second half of my talk, I'm going to dive into important CMC considerations for human gene therapies that incorporate human genome editing. So let's start with a basic definition, how we define genome editing. So it is the process by which DNA is inserted, deleted, or replaced in a site-specific manner in the human genome using nucleases or other non-nuclease methods. So nucleases create a specific site strand break, which is repaired by inherent cellular mechanisms. Those two mechanisms are primarily non-homologous end joining, which I'll refer to short as NHEJ, and homology-directed repair, or HDR. And I'll get more details into how they work in a few slides. So genome editing technologies, right? So you can see depictions of 4 different types of genome editing down the right-hand side of the slide. The first one, the top, is a meganuclease, which is an engineered version of homing endonucleases that contain both the nuclease activity and targeting activity. Second, so zinc fingers are one of the most common DNA binding motifs in biology. So by combining them with a nuclease domain, in this case FOC1, zinc finger nucleases are fusions of DNA cleavage domains from a FOC1 and endonuclease activity with zinc finger proteins. They generally are combined or engineered to recognize between 9 and 18 base pairs of DNA. The third one on the list today is TALEN. So tail proteins were identified from plant pathogenic bacteria. And again, they're fused to FOC1. They recognize a single base pair each. Again, they're engineered to recognize close to 20 base pairs for their nuclease activity. The last one on the list is probably one of the more common ones with a very difficult name. So clustered regulatory interspace short palindromic repeats, or CRISPR for short, was discovered as a bacterial defense against invading viruses. So CRISPR ribonucleoproteins will associate with PAM motifs throughout the genome, which is a 3-nucleotide sequence, and then interrogate the sequence directly upstream of that for complementarity to their guide RNA. So a guide RNA binds to the Cas9 protein and has a spacer sequence that should complement the DNA genome and the target site. Upon DNA RNA duplex formation at the target site, the Cas protein actually undergoes a conformational change that positions its 2 nuclease domains to create a double strand break in the DNA. So nuclease base editing, this is a slide to sort of show the mechanism of action. Like I said previously, it uses inherent DNA repair machinery. The first is called non-homologous end joining. This is basically the joining of 2 blunt ends of DNA that often results in incorporation of errors, such as small deletions or insertions of a few base pairs. Homology-directed repair uses a DNA donor template with homology around the target site, which is used to incorporate genome edits in the DNA template into the genome. So we also have many variations on these nuclease-based editing methods. The first are called base editors, and the second, prime editors. They use additional catalytic activity domains to incorporate genome edits. Base editors are generally incorporated with a deaminase domain, are designed for single base pair conversions, and prime editors use a special guide RNA called a PEG RNA, along with a reverse transcriptase domain. I'll note that both of these actually use what's called a Cas9 nickase, which is a Cas9 protein that's been engineered so that one of the nuclease domains is inactive, and so it only nicks a single strand of the DNA and does not cause a double strand break. And I'm going to go into a few more details on the mechanism of action for these 2 types of genome editors. So the 2 basic base editors are cytosine deaminases and adenosine deaminases, and they work by a very similar mechanism to create a single base pair change. The cytosine deaminase actually targets a C in the window and converts it to a uridine, which then by cellular repair machinery is converted to a tyrosine. So you get the C to T transition. Likewise, adenine base editors convert an A to inosine, which is then changed to guanine. And we get the A to G conversion. For prime editor mechanism of actions, this is a bit more complicated. So the nickase, in this case, nicks a single strand of the RNA, which is then used to prime the PEG RNA. The PEG RNA is then activated by the reverse transcriptase to insert new DNA sequence onto the end of the DNA strand, creating a flap with new DNA sequence. Via, again, cellular repair mechanisms, that flap can either be incorporated or removed from the target site. You note that if the edit is actually removed, that now again becomes a template for prime editing, which subsequently could be changed to incorporate the desired genome edit. So here are some examples of the ways these different editors could be used to provide a therapeutic benefit. So NHEJ can be used, for example, to cause a gene disruption for different pathogenic genes by causing small indels inside the gene that disrupt transcription of that gene. You can see examples of that in A and B in this figure. Homologous directed repair can be used to insert a new gene into a target locus that has a mutant gene, or add an additional therapeutic gene into a safe harbor locus, as depicted in figures C and D on this slide. So there are many potential therapeutic applications for genome editing. I'm going to cover a few of them on this slide. The first one, of course, is hematologic disorders such as SCID or sickle cell disease. And in fact, last year, the first genome editing product was actually approved for commercial use to treat sickle cell disease. They can also be used to treat neuromuscular disorders, ocular diseases, skin diseases, lysosomal storage disorders, viral infections, and even cancer in some cases. So that brings us to our first challenge question of the day. So which technology uses a nickase? You have choice A, meganucleases, B, prime editors, C, talons, or D, CRISPR-Cas9. So I want to give everybody a moment to think about your answer. This would be a good time for me to take a drink. Does everybody have their answer? So the answer, of course, is prime editors. They use a nickase to nick a single strand of DNA, which is then used to prime the PEG RNA and write new DNA into the sequence. So that brings us to my second half of the talk, where I'll be discussing CMC considerations for gene therapy products that incorporate genome editing. So we sort of divide genome editing products into two main categories. Genome editing products include both directly administered, so we call those in vivo administered products when they're administered directly to the patient. Those are generally the components or machinery for genome editing that can be delivered by viral particles or non-viral based methods such as lipid nanoparticles. We also have ex vivo modified cellular products where cells can be removed from the patient, grown in a dish or manufactured in a dish. The genome editing components are then added to affect the given edit, and then the genetically modified cells can be given back to the patient in the clinic. So there are many considerations that you should think about when you're developing a genome editing product. Those include the type and degree of the modification needed, the mechanism of DNA sequence change that you choose to employ, the product design and delivery method, so in vivo versus ex vivo modified cells. Delivery methods can include lots on this list, such as like viral vectors and nanoparticles, as I mentioned, even plasmid, mRNA, and proteins. Of course, the optimization of the genome editing components expression is very important to get the desired genome edit. And on and off-target editing studies need to be done to assess and model available and appropriate for off-target editing and on-target editing, right? So on-target editing, you'll need to monitor either sequence changes, expression of the gene, or the biological downstream function of the desired edit. Success of any given strategy will depend with the ease with which the therapeutic genomic modification is achieved, the efficacy of the delivery of the genome editing components to the target cell type, and the fitness of the edited cells themselves. And lastly, of course, you need to consider the clinical trial design, patient monitoring, and long-term follow-up. So the regulation of genome editing products is based on a science approach consideration. That includes a characterization of product mechanism of action, the safety attributes, using current knowledge and tools in the field. It also includes a benefit risk analysis, such as the potential to correct the genetic causes of the disease, the risk of unintended genome modification, and the unknown long-term effects of on or off-target genome editing. We also consider the severity of the disease and the potential for improved quality of life. So ensuring genome editing product quality really starts with suitable qualification of starting materials and components, a well-defined process and process controls, and appropriate product testing and characterization. So genome editing components we define as the nuclease, any targeting elements, and donor templates where appropriate. We consider these components to be drug substances when they are formulated into the final drug product or a part of the active pharmaceutical ingredient, or API, that is directly administered to perform the genome editing in vivo in the patient. We consider them critical components when they are used to perform genome editing in cells ex vivo, and the autologous or allogeneic cells themselves are actually the drug product. Due to their criticality, if components are modified during the product life cycle, comparability studies may be necessary. So to get a little bit more into genome editing components consideration, so you should provide a detailed description of how the components are designed, manufactured, and tested, and all that should be included in your IND submission. In most cases, components should be manufactured according to current good manufacturing practices, or CGMPs for short. You can refer to the FDA guidance for industry on CGMPs for phase 1 investigational drugs for guidance on how these components should be manufactured using CGMPs, even for early phase clinical studies. And of course, full CGMPs are expected for BLA supporting clinical trials and licensure. Genome editing components should also be tested for safety, identity, purity, activity, and residuals based on their manufacturing process. And specifications should be determined based on manufacturing experience and what has been shown to be safe and effective in preclinical or clinical studies. And of course, the stability of genome editing components should be assessed. So genome editing drug product testing should also be tested for safety, identity, purity, and potency. And again, you should be setting specifications based on your manufacturing experience and what has been shown to be safe and effective in preclinical studies. We have a few additional concerns for ex vivo modified cell products, and those include characterizing the presence of residual genome editing components in the cellular product before delivery, the need to test each batch for off-target modifications or translocations or other chromosomal abnormalities. And of course, allogeneic products need to be thoroughly characterized to ensure product safety. All these are generally considered on a case-by-case basis, depending on your product. So I want to go into a little bit human genome editing product safety concerns. So the first, of course, is off-target genome editing. So you need to consider the type and sensitivity of off-target screening methods you will use, as well as an additional concern for in vivo products, looking at off-target cells or tissues, depending on the results of such as a biodistribution study in your preclinical data. Unintended biological consequences of on-target editing need to also be considered, such as mutagenesis from imprecise DNA repair following on-target editing. And of course, you need to assess the adverse effects of genomic DNA cleavage at on and off-target sites, such as chromosomal translocations, inversions, or other abnormalities. For all three of these safety concerns, it's important to detect potentially deleterious events even at low frequency. The last concern listed on this slide is immunogenicity. So that could be to the genome editing products components themselves, the editing outcome, or the delivery system. So in assessing the potential for unintended genomic modifications, you should look at appropriate methods for predicting and identifying unintended genomic modifications. We recommend the use of orthogonal methods that best suit your product. Some modalities may require additional specific analyses, such as base editors or epigenome editors. You should provide analysis details, including experimental parameters and sequencing depth and quality, as well as provide an annotated list of the unintended genomic modifications observed. You also need to account for genomic variation between individual human subjects. You should use appropriate databases based on the intended patient population. You should set variant frequency cutoffs based on a risk assessment and use methods for confirmatory testing as needed. You should also determine the biological impact, where possible, of the identified unintended genomic modifications. We note that not all off-target genome modifications will necessarily lead to adverse biological consequences, and we acknowledge that addressing the potential for downstream biological impact is a significant challenge. So this chart lists a few of the most common methods for identifying off-target modifications. It's important to use methods that best suit your product, using a combination of in silico and in vitro methods that both nominate potential off-target sites and test or confirm those targets. In vitro testing may be performed in surrogate cells, but you should consider the comparability of the target cells and how chromatin structure or editing may be different. The rapid evolution of these tools has certainly improved our ability to evaluate the safety of therapies based on genome editing. However, due to the lack of complete coverage with any one method, we recommend the use of orthogonal methods for evaluating off-target genome editing. So I'm going to discuss a little bit about methods for identifying chromosomal modifications. Those should include in silico modeling, as well as cellular approaches such as image-based genome analysis, like FISH or karyotyping, as well as biochemical approaches like unidirectional sequencing. So I'd like to provide a summary of this section of the talk. So genome editing products include directly administered or in vivo products and ex vivo modified cellular products. Genome edited components can be considered drug substances or critical components, depending on their use. In both cases, they should all be tested for safety, identity, purity, activity, and residuals and stability as necessary. And of course, they should be manufactured under phase-appropriate cGMPs. Genome editing products should be thoroughly tested depending on the product type. So that brings us to our second challenge question, and that is, which of the following statements is not true? So A, genome editing components can be considered drug substances or critical components. B, detailed descriptions of how the genome editing components are designed, manufactured, and tested should be included in an IND. C, in most cases, genome editing components do not need to be manufactured according to CGMPs. Or D, if genome editing components are modified during the product life cycle, comparability studies may be necessary. Please take a moment to consider your answer. So of course, the answer is C, as this is a false statement. Genome editing components should always be manufactured according to phase-appropriate CGMPs. So before I end my talk today, I wanted to provide you with some additional guidance. First, the FDA has recently released a guidance for industry on human gene therapy products that incorporate human genome editing. It provides recommendations for information that should be provided in an IND application to assess the safety and quality of investigational genome editing products, and it can be found on the FDA website. Additionally, the FDA earlier this year recorded a webinar that covered questions and answers based on the genome editing guidance. I'd also like to mention a few early communications with CBER OTP, which include interact meetings, which are non-binding, informal discussions between non-clinical review disciplines, such as pharmacology, toxicology, and CMC, and the sponsor. And this initial targeted discussion is for specific issues after obtaining preliminary data from pilot studies, but prior to conducting extensive animal studies. Before submitting your IND, you can schedule a pre-IND meeting, which is a non-binding but formal meeting between the FDA and the sponsor. Meeting packages should include summary data of sound scientific principles used to support the use of your specific product in a specific patient population. And of course, guidance, again, for these are located on the FDA website. The last one I wanted to mention is CAT meetings, which are conducted with the CBER Advanced Technologies team. These are early discussions involving novel manufacturing processes or analytical methods. So my last slide in the talk covers some CBER contact information, including my contact information, as well as the regulatory OTP mainline, and references for CBER OTP regulatory processes and interactions with CBER OTP, as well as the OTP Learn webinar series. So that concludes my talk for today. And I think Gopa will be our next speaker.
Thank you, Brian. That was a wonderful talk. So I have two announcements. The first is that for the in-person attendees, a cell phone has been found and handed into a registration desk. So if you're missing your cell phone, please go off and reclaim it. Secondly, I'd like to remind our online audience to use the chat pod to enter any questions, because we're really interested in hearing what people who are attending virtually have, what questions they may have. So now I'd like to hand it over to Gopa for her talk on facilitating development of gene therapies for rare diseases. Go ahead. Thank you.
So good morning, everyone, here and also online, and also good afternoon or good evening if we have any colleagues joining from other parts of the world. I'd really like to thank the meeting organizers for the opportunity to discuss some of the work that we are doing in CBER to facilitate the development of gene therapies for rare and very rare diseases. So during the course of our discussion today, I'm going to be discussing, what we mean by an individualized or a bespoke gene therapy, and then talk about a public-private partnership called the Bespoke Gene Therapy Consortium, which is an area where CBER has been very actively engaged. And then I'll share a few thoughts on regulatory streamlining when we're working in the rare disease space and the importance of global collaboration. So, I just like to start by what do we mean by rare? So the Merriam-Webster dictionary defines rare as something that's seldom occurring or found or uncommon. But in the rare disease space, the definition of a rare disease is a disease that affects fewer than 200,000 people in the United States, which is about 1 in every 1,500. There are now they're saying 10,000 different rare diseases affecting a total of 25 to 30 million Americans and many more patients globally. And so this really represents a significant unmet medical need. And if you think about the number of people who are affected by rare diseases, having a rare disease is not really that rare. It impacts a lot of people. So many of the diseases are serious and life-threatening, and many affect children, resulting in significant morbidity and early death. And approximately 60 to 70% of the rare diseases are single gene disorders that have the potential to be treated with gene therapies. And that is delivering safe and effective gene therapies to those in need is a global priority to alleviate the suffering from various serious diseases. And gene therapies, research in gene therapies is one of the fastest growing areas of research, with gene therapies for many diseases, both common and rare, under clinical development. So in this slide, what I've tried to demonstrate is really the increase in the amount of research that's going on in this field, as demonstrated by the increase in the number of INDs that are being submitted to FDA. And this goes through 2021, but that trend is continuing. So it really is a significant research. There is a significant research investment in cell and gene therapy development. On this slide, I've listed the currently US approved gene therapies. And the point I'd like to make is that, 2017 really was a breakthrough year with the licensure of 3 different gene therapies. But then in 2019 and 2020, there was approval of a single gene therapy each year. But as you can see in 2022, 2023, and 2024, there's been a great increase with 4 to 5 products being licensed each year. And this trend is going to continue. And what I've provided here is a website where if you want information about any of these gene therapies, this information is available online. So I would like to now start by defining what do we mean by individualized therapies and why do we need to think differently about the development, licensure, and access to these products. So individualized therapies are based on engineering a product aimed at the specific mechanism underlying a patient or a small group of patients' illness. So that is that the therapeutic product is engineered specifically for a given patient or a small group of patients. And in this regard, it's really different from the majority of the products that we regulate at CBER. So some examples of individualized therapeutics are antisense oligonucleotides, or ASOs, directly administered gene therapy or gene editing for monogenic diseases, genetically modified cellular therapies, and genetically engineered phages for multi-drug resistant infection. ASOs are regulated by our sister center, CDER, but the other 3 product areas are regulated by CBER. So I've included this slide just to emphasize that individualized therapeutic products really present some unique challenges because they do not fit the traditional paradigm for manufacturing and clinical development. And so we really need to think differently about development and regulation of these products while still assuring that the standards for quality, safety, and efficacy are met. And that's really the challenge that we're trying to address. So although gene therapies hold tremendous potential, and we've already seen that from some of the products that are currently licensed, there are challenges. And one such area is in manufacturing. So the current manufacturing constraints present what we can describe as like a Goldilocks phenomenon. So, commercial solutions are not available for diseases that affect very large or very small populations, but there is a sweet spot in the middle where commercial viability relies on products that are going to treat between 100 and a few thousand individuals. So I'm going to focus on the one end of the spectrum, which is how do we make gene therapies available for diseases that affect very small populations? And by very small populations, I'm talking about treating 1 to 100 people in the US a year. So I'm going to discuss the work that we've been doing with the Bespoke Gene Therapy Consortium, or the BGTC, which is a public-private partnership that's working towards streamlining gene therapy development for rare disorders with the goal of making the process more efficient and less costly. The BGTC is a collaboration between the Foundation for the NIH and CATS, as well as FDA CBER. So the leadership of the BGTC is through these organizations, but participation involves many industry partners as well as nonprofit organizations that are active in the rare disease space. And the idea is to work in the pre-competitive area to promote collaboration, information sharing, and really to stimulate innovation and ultimately to facilitate access of these products for the patients who need them. So the objective of the BGTC is really to advance developing gene therapies for very small populations. And in order to do that, all of us, regulators, researchers, patient groups, everyone needs to start thinking outside the box. So the BGTC pilot, will focus on clinical trials for disorders that have such low prevalence that there's no current commercial interest. There's no intention to be competing with industry. If industry is working on a particular disease area, that's great. What the BGTC is trying to do is to fill a gap that currently exists, which is development of gene therapies for very rare disorders. One of the major goals is to work on streamlining the regulatory requirements to facilitate product development. So for example, through establishment of master files for standard vectors, and also looking at opportunities where there can be leveraging of data from one application to another when it's appropriate to do so. And although the BGTC is working in the very rare disease space, we anticipate that the program will have broader impact for more common disorders with higher prevalence, because the learnings from the BGTC experience can be applied to other gene therapies also. So this is a slide that's a schematic that shows some of the goals of the consortium. But the bottom line that I want to state here is that one of the objectives is to standardize things as much as possible. To standardize having a suite of AAV vectors that can be used for development, to standardize the testing that's done, and to standardize the clinical delivery methods and the clinical protocols. And by standardizing a process, that can then be leveraged for development of other therapeutic products. So the pilot will really utilize or leverage existing manufacturing capacity and experience. So industry partners or CDMOs that have experience manufacturing AAV gene therapies will be manufacturing the gene therapy products for the pilots using their standard manufacturing process. As I mentioned, the testing will be standardized to the extent possible. And a decision was made early in the program to run these clinical trials sequentially rather than concurrently, so that the lessons learned from the early trials can then be applied to the later trials, including input from the FDA that's received through interactor pre-IND meetings. And I just want to really take a minute to talk about the U.S. government's commitment to publicly share the information from the BGTC. So the NIH will be the IND holder for 5 of the clinical trials that are funded by the BGTC. And as the IND holder or as the sponsor, they have the right to share the information publicly if they wish. We as FDA cannot do that. You know, information that comes into the FDA is confidential, and we don't have the ability to share that information publicly. However, the sponsor can do it. And so the NIH has made a commitment to publicly share IND submissions and official FDA communications for these 5 trials, and to place the data from the clinical trials in the public domain. And this really is intended to facilitate the ability of other researchers to leverage what's been done and the lessons learned for development of other gene therapies for rare diseases. And although the last bullet point is not an NIH-specific activity, it's actually a BGTC effort. The BGTC is developing what they're calling a playbook, which is a guide for clinical development of AAV gene therapies. And version 1 has already been published, and its focus is primarily on navigating the regulatory path from the early stages to conduct first-in-human clinical trials. So it was really exciting after 2 years of planning to have the Bespoke Gene Therapy Consortium officially launch on October the 27th, 2021. And I've included some clips from the joint FDA-NIH press release, which includes a quote from Peter Marks, the Center Director, CBER Center Director, highlighting FDA's commitment to advance gene therapies to meet the needs of patients with rare diseases. This is clearly one of CBER's priorities. So on this slide, I've listed the current membership of the BGTC, and you can see that there's a broad range of stakeholders that have signed on to be part of the consortium, including multiple NIH institutes, and it's primarily being led by NCATS, the National Center for Advancing Translational Sciences. Many industry partners, many of them are very active in the gene therapy field, and nonprofit organizations that are active in the rare disease space. The expertise within the consortium is really impressive. There's been investment of over $100 million in the program from both the public and private sectors for the 5 to 6 years that the pilot is expected to run. So now I'm going to talk about the work that's been done since the launch of this public-private partnership in October 2021. Following the launch, there were several sub-teams that were established consisting of subject matter experts from the membership of the BGTC. And as you saw in the earlier slide, the membership of the BGTC includes many of the major players in the field. So there's really a deep breadth of expertise within the consortium. And each sub-team has taken on specific areas of work that they want to advance. I'm going to briefly focus on some of the work that's been led by the clinical, manufacturing, and preclinical toxicology subteams, but the other subteams have also taken on significant activities, but in the interest of time, I'm not going to be able to cover them. I want to spend a little bit of time on this issue of disease selection for the pilot. You know, as I mentioned, there are over 10,000 rare diseases. Every single disease is important, and finding treatments for all of these diseases is important. But there were some very clear criteria that were set in working towards selecting the diseases that would be part of the BGTC pilot. Some of the characteristics that the BGTC was seeking are listed on the right-hand side of this slide. So there needed to be demonstration of a clear monogenetic cause for the disease. So in other words, a correlation between a defective disease, defective gene being the cause of a disease. And it also needed to be a disease that was amenable to treatment with AAV gene therapy, because the BGTC is really focused on AAV gene therapy treatment. There should be availability of an animal model with the relevant phenotype. And as I mentioned, the BGTC's focus is on diseases where there's no commercial interest. Another consideration is the availability of sufficient information to run a successful clinical trial. For example, availability of natural history data would be incredibly valuable. So that was a factor that was taken into consideration. And one practical factor is management of requirements for testing and follow-up. So for example, if a treatment effect could be determined in a relatively short period of time, that was something that was of interest because some treatments may take 15, 20 years to see if it's effective. And so for the purposes of the pilot, that was one of the considerations. And another important factor was the availability of an active patient group that could help with enrollment in the trial. So when we're talking about very rare diseases, we're talking about a handful of people living throughout the United States. And so we really wanted to have an active patient group that could identify the patients for recruiting them for clinical trials. And there were other considerations, but I just highlighted some of the more critical ones. So this slide shows the process that the FNIH went through to identify the diseases that would be part of the pilot. So FNIH posted an expression of interest for diseases and received 63 nominations for 62 different diseases, and that alone showed the great interest in the program. These applications were reviewed by the clinical sub-team, and it was down-selected down to 14 candidates based on the selection criteria that I discussed. And then there was outreach to these candidates to submit full clinical proposals. And then those full clinical proposals were reviewed by the clinical and also the non-clinical or the preclinical sub-teams, and the 8 diseases were selected. And so on this slide, I've listed the 8 diseases that are part of the BGTC portfolio. These were selected based on the strength of the applications that were submitted, taking into consideration the criteria that I spoke of, and also the relative readiness of these programs to proceed to first-in-human clinical trials. So there are 3 ocular diseases, 3 neurological diseases, and 2 systemic diseases. So there's a nice, and this just happened organically. These were the applications that really scored the highest. And so there's a nice distribution across different diseases in target audience, target organs. And so in line with this, the gene therapies in the pilot will utilize several different AAV serotypes because the different serotypes target different organs. And so in this slide, I just want to note a few things. One is that 5 of the 8 clinical programs that are part of the BGTC will be funded by the BGTC. Those are the ones that are in pink and also propionic acidemia. The remaining 3 that are highlighted in green are going to be funded by the California Institute of Regenerative Medicine. But the idea of those programs are going to be very much in line with the objectives of the BGTC. It's just that the funding will be separate. And I've highlighted propionic acidemia because it's the only clinical trial that will be conducted at the NIH Clinical Center. All the others will be conducted at external non-NIH sites. So I just want to briefly touch on 2 significant activities that have been taken on by the manufacturing and preclinical subteams. So the manufacturing subteam worked together to develop a proposal for what they defined to be the set of minimum critical quality attributes for gene therapies for very small populations. They submitted that proposal to the FDA, and there was a meeting with technical experts with my colleagues who are in the Office of Therapeutic Products. And it was an informal discussion. It was a scientific discussion. It was not product specific, and it was not binding, but it was incredibly helpful for providing some early input to the BGTC on what is the FDA looking for in terms of the testing requirements. That was so successful that the preclinical and toxicology subteam wanted to do something similar. So they developed a proposal for a minimum toxicology testing package for AAV gene therapies for very small populations. And this, then they looked at it from the perspective of what's needed for ocular administration, for neurological and systemic. And similarly, they had a meeting with the FDA, and the discussion was very valuable in providing some early input before the preclinical and toxicology testing is done for some of these programs. And all of these activities are really focused, again, on the purpose of the BGTC, which is to streamline testing and promote a platform approach for AAV gene therapies for rare and very rare diseases. I'm not going to get into this slide because it's very busy. I only want to make one point, and I go back to stating the tremendous amount of expertise coming in from the members of the BGTC. And all of that expertise is being used, and the principal investigators of these 8 programs are greatly benefiting from the input that they are getting from the expertise available. Each of these 8 diseases has a cross-functional team bringing in expertise from all the sub-teams, and it's, I guess the lesson learned from this is collaboration really does tremendously impact the ability to move programs forward quickly. I spoke to the BGTC playbook in version 1.0 being available. The focus of this version is on the navigation of the regulatory pathway. There are resources available at the FDA. There are different types of meetings. Brian spoke to some of them. And so the idea here is to help investigators navigate that. But with experience within the BGTC, as these clinical programs progress, the playbook is going to be updated with more granular guidance on development of AAV gene therapies for rare diseases. So now I will focus the next few minutes on the approaches to providing regulatory flexibility to support the development of gene therapies for rare diseases. There are some important considerations for clinical development of individualized therapeutics. For example, what quantity of supportive preclinical evidence do you need prior to treating a patient? And how are we going to know if the therapeutic product is effective? So it's important to really understand the relevant disease-specific clinical information that should be captured when the patients are treated. In addition, we need to assure that the manufacturing process generates a product that's fit for purpose. And the last bullet point, I think, is really important because we have to start thinking about a sustainable way to maintain access for patients. These are all important considerations when we start thinking about how do we facilitate development of individualized therapeutics for different diseases. But you must also consider the aspect of sustainability and patient access. There are potential opportunities for regulatory streamlining to support these efforts, and these are areas where FDA obviously will play a very significant and critical role. Master files can be used for providing manufacturing information to support IND applications if the same vector is being used for development of multiple products. One objective will be to see how much information can be leveraged based on past experience with the same vector or similar gene therapy product. This is obviously an area that we're putting a lot of thought into because it's so critical, especially in the rare disease space. And then looking ahead, adaptive clinical trials could be considered. And then if any of these investigational products show clinical benefit, we need to think about the path to licensure. But the BGTC is in the early stages, and the focus really at the moment is on the first 2 bullet points. And I just put this to remind ourselves that when we're speaking about rare and very rare diseases, we really have to think innovatively because the standard processes are not applicable. The number of patients is too small. And so if we're going to make these products available for patients, we really have to think about new possibilities. So what is the current state? Well, many gene therapies for rare disorders are produced as one-offs in academic laboratories or small corporate entities. There's a lot of research going on, but it's in forums that where the researchers may not have a lot of experience working with the FDA. And the current regulatory process does not leverage information to expedite the production of potentially transformative treatments. So a regulatory program that allows leveraging of non-clinical and manufacturing data from one application to another can facilitate product development and promote access. And so what I've just described is the concept of originator product and offshoot products, which is illustrated here on this slide. So here are 4 products, the original and 3 offshoot products that use the same vector backbone, but differ in the transgene insert. And the question is, how much information can you borrow from one situation to another? And this is something that's going to require considerable discussion between FDA and relevant stakeholders, including industry partners, to develop a regulatory framework that can leverage existing knowledge to advance the development of bespoke gene therapies. Now, at the FDA, I have to emphasize that we're a science-based organization, and any decisions that we make are based on science and data. So as we look at the issue of leveraging from one application to another, we have to keep in mind what are the data that allow us to do that. But it is something that is critically important when we're talking about gene therapies for rare diseases. And it's hoped that the BGTC clinical trials will contribute to data that will help us address some of these questions. So for the last few minutes, I want to talk a little bit about our global engagement and why global collaboration is especially important when we're talking about product development for rare diseases. So research and development in gene therapies is occurring primarily in high-income countries, and that's not surprising. At least that's the case in the present situation. And commercial viability currently requires administration of 100 to 200 treatments per year. But for very rare diseases, there may be only a handful of patients in any given country. So there could be a handful of patients in the US, handful in Europe, a handful in Japan. And so any one country may not have enough patients in order to have, commercial viability in terms of industry wanting to get engaged in product development. However, if you market across high-income countries, that could get the number of patients high enough to promote commercial viability. And regulatory harmonization among the high-income countries really could facilitate commercial development. So as noted, clinical trials for gene therapies and licensure of gene therapies is currently limited to high-income countries, those with more experienced regulatory authorities. However, patients in low and middle-income countries may actually benefit the most from gene therapies because they often lack access to supportive care. And so a gene therapy that provides long-term benefits or potentially a cure can be transformational for patients. For patients globally, but in particular in low and middle-income countries. And regulatory harmonization could facilitate commercial development in high-income countries, and approvals in high-income countries could then be leveraged to provide access to patients in low and middle-income countries through the practice of reliance. So I emphasize this because the work that we do here and the licensures and access in high-income countries could potentially open the door for these products being used in low and middle-income countries. So I want to talk about a few areas where CBER is engaged in the international arena to facilitate the development of cell and gene therapies, especially for rare diseases. So one new initiative is the COGENT pilot. COGENT stands for Collaboration on Gene Therapies, and this is a global pilot. It's an effort to encourage sponsors with global development programs that include the US to invite other regulators to early stage meetings, such as INTERACT meetings or pre-IND meetings. The global pilot is really intended to facilitate collaborations in internal meetings and as well as review of applications with select partners. Obviously, this will have to be done under strict confidentiality. But the goal is really to increase efficiency of the regulatory process, to reduce the time and the cost both for the regulatory agencies as well as the sponsors for assessment of these products. Another area where we're very actively engaged is in harmonizing the regulatory approach that's taken in high-income countries. So we're in continuous dialogue with our regulatory counterparts at EMA, PMDA, which is in Japan, Health Canada, et cetera, and collaborations through ICH and other international venues. When there's regulatory harmonization, it really makes it much easier for product developers and it promotes faster development and access. So this has been a high priority for us. And finally, I'd like to touch on our work with the World Health Organization. The reality is that the vast majority of countries in the world do not have a regulatory framework for cell and gene therapies. And they know that these products, at least cell therapies, are coming across the border and their patients are utilizing them without any oversight. So this has been a priority. And we've been working with the WHO and regulators in other regions of the world to develop a WHO document on considerations for a regulatory framework for cell tissue and gene therapies. It's a high-level document that really focuses on the important principles that one needs to be thinking about when establishing a regulatory framework, and more specifically, a risk-based framework for evaluation of cell and gene therapies. And I've included a link to the document if anyone is interested in looking at it further. This was adopted as a WHO international standard in March of 2023, and we continue to collaborate with the WHO to support low and middle income countries in implementing this considerations document and in strengthening regulatory capacity in this area. So to summarize briefly, delivering safe and effective gene therapies to those in need should be a global priority. To alleviate the suffering from various serious and often rare diseases. Patient suffering here is the same as patient suffering elsewhere in the world. And so we really need to take a global perspective on this issue. And global regulatory convergence in high-income countries could promote commercialization and pave the way for use of gene therapies in low and middle-income countries. And I'd like to just I've included a quote here from Peter Marks, who's the CBER Center Director, that says, CBER aims to make 2024 a breakthrough year addressing key challenges to the development of cell and gene therapies, especially for rare disorders. CBER is very much invested in this area. And as noted, it's been a priority in the past, but most especially this year. So I have proposed two challenge questions. The first one is looking at, what is an individualized therapeutic? So an individualized therapeutic is a treatment designed for A, only one patient, B, for all patients with the same rare disorder, C, for one or a few patients with the same rare monogenic disorder, and D, for a class of rare diseases, for example, all monogenic neurological disorders. So mark your response in your head. Okay, the answer is C, for one or a few patients with the same rare monogenic disorder. And then for challenge question number 2, it's really focusing on what actions facilitate development and access to gene therapies for rare diseases. The options are A, regulatory streamlining and harmonization of requirements, B, collaboration among regulatory authorities, including collaborative reviews, C, establishment of regulatory framework in low and middle income countries, which includes the legal authority to practice reliance on the work of others, and D, all of the above. This one is too easy. I'm only going to give 2 seconds. The answer is all of the above. These are all actions that will help facilitate development of gene therapies for rare diseases. So on this slide, I've just listed some resources if anybody's interested in looking at more information on the BGTC. I've also listed this as a resource. This was really the very last thing we did at FDA before things shut down for the pandemic. This was a workshop in, I believe, early March 2020. And it was focused on facilitating end-to-end development of individualized therapeutics. There's a link to the workshop at the bottom, and a recording is still available along with the transcript. So I just want to share a few closing thoughts. When it comes to rare diseases, it really does take a village. With over 10,000 rare diseases affecting 25 to 30 million people in the US, there really is a significant unmet medical need for these patients. These biological products hold tremendous potential, but there are also challenges. And so collaboration among the many stakeholders, including researchers, manufacturers, those working in clinical development, and those involved with patient groups and patient access. All of these entities are important for advancing development of these treatments, which can truly be transformational. And so I just want to conclude by emphasizing that we all have a role. And if your research interests relate to any one of these 10,000 plus rare diseases affecting individuals in the US and globally, you can play a role. Work collaboratively with other stakeholders that have similar interests. By working together, we can get there faster. And I just want to conclude by saying that the time for doing that is now, because patients are waiting. So thank you very much for your interest in this topic and for taking the time to join today, and I'd be happy to take questions during the panel session. Thank you.
For today, and we're ready to have a slightly extended question and answer session, and the microphones are open. So please go ahead and start with the questions.
Hello, very nice presentations. I have a quick question regarding the COGEN-T program. It was nice to see you mention it. I've heard it mentioned by other leadership within CBER, like Peter Marks and Nicole Verdun. I like to find more information. I tried looking on the FDA website. It was really difficult to find more. Can you speak a little bit more to how a sponsor who would be interested in collaborating with different regulatory agencies to facilitate the process? I understand from your talk and previous meetings that sponsors can invite whoever they want. However, there seems to be a few logistical challenges involved with trying to facilitate that. I know EMA and other regulators have true simultaneous review where you get opinions from multiple regulators. Is that the vision for the COGEN-T program? And is there a way to actually submit those applications, or how is CBER thinking about actually executing on that?
Is this on? Oh, great. Really good question. And I will say that I don't have all the answers because this is very early in the process. Our initial effort is going to be working with the EMA. And the idea is really to look at applications that are at a stage where it's similar with FDA and EMA. Because this is a pilot. And so we just need to see if the process works. So at this moment, it's not so much that sponsors are going to be submitting applications. Down the road, that is the vision if the pilot shows promise. But right now we're looking at to see whether the process would work. And our starting point is looking at applications that are in with EMA as well as FDA and what would be a good candidate. So it's in the very early stages.
So if a sponsor wanted to participate in the pilot, would sponsors need to reach out to OTP or OCI? Like, how would they actually participate in the pilot?
Yeah, at the moment, we're working with the EMA to see what may be a good candidate just as a starting point. But the vision is that if this shows promise, and there's good reason to think that this would be a good thing to do just based on our past experience, then the process would be formalized further with opportunities for sponsors too.
All right, thank you.
Thank you. Can we have the next question at the microphone, please?
So you mentioned a discussion of what you called a original product and then secondary products with gene therapy using the same backbone with different inserts. But that sounds a lot like there are other platform technologies that are being developed out there. Is there any crosstalk between this looking at extremely rare diseases and then this sounds a lot like even nucleic acid vaccines, which of course are being used in much larger populations?
I'm going to turn it over to my colleagues in OTP, but let me just touch on the question that you asked. You're absolutely right that the idea of platform technologies is not unique to gene therapies or rare diseases, but it's absolutely critical for product development for rare diseases. But I think it was just yesterday the guidance was is it today? Yeah, the guidance was published for the platform technology designation program, providing guidance on what the thinking is behind that. But that's the designation program. When I am engaging with stakeholders in the rare disease space, actually the idea of what is a platform technology is something that's being asked more broadly, and that's obviously something that's under discussion internally also. But yes, it's not a new concept, but it is one that's critical for advancing therapies for rare diseases. Is there anything?
I don't have anything to add.
Okay, I think we'll do one more question at the microphone, and then we'll go to the online questions.
Sure. This question is for Brian. When we're looking at assays to detect off-target edits, what are your recommendations if we do find an off-target edit, but you don't know where it's coming from, right? I mean, it can happen naturally, and then if you report, there's certainly fallout from that. So, what are your recommendations when working on an assay to develop off-target edits?
Yeah, so as I mentioned in my talk, it's really a sort of a combination approach. So you should have methods, sort of in silico methods or methods that nominate potential off targets. And then you should have in vitro methods that sort of test or confirm those off targets. And certainly, as I said in my talk, there you may discover that you have valid or real off targets based on your targeting system. And so the idea is that you try and, as best as you can, sort of provide a justification or rationale of what the biological effects of those off targets might be. And we understand that can be difficult sometimes, but you should, try to address that as best you can in an IND submission as far as the justifications.
Thank you. Can we have our first online question, please?
All right, this first question is going to Brian. Can you please provide context on what off-target assessment may be needed for early studies, example, to support IND clearance, versus what off-target analysis may be provided later in development?
So really the off-target assessment is really based on the safety of the product. And so even for early phase clinical studies, you need to sort of establish the safety of the product. And so most of those sort of off-target analyses or methods really need to be done up front in the preclinical stage of product development. So they be submitted in the original IND submission. Certainly for additional advice on your proposed methods for an off-target analysis, pre-IND meeting would be the appropriate time to discuss that with the agency. You can go over your plans or methods or even results if you've already done some analyses with the agency, and we can provide you feedback for your IND submission.
Okay, thank you. We'll take one more online question and then back to a microphone.
Okay, so this question is for Gopa. Regarding projects to be selected, how is the criteria of lack of commercial interest measured?
So if I understand the question correctly, why did we focus on products that don't have any commercial interest?
I think the question is, how do you select products? How do you determine whether there's a lack of commercial interest?
Okay, thank you. Really, there was a lot of research done. I mean, the members of the BGTC are really quite well connected in terms of, having an awareness of what products are under development. I know not all companies share everything, but there is a good, there was research done in terms of, is there any evidence that there is, activity by companies in development of some of these products, and so that was the way it was determined. Now, one point I want to make is that, and I always emphasize this, we say no current commercial interest. And why do we emphasize that? Because if what the BGTC is doing is successful, and if there are ways to incentivize development of gene therapies for these very rare diseases, the hope is that some of these diseases are of no commercial interest today, there will be commercial interest in the future. So that is really one of the ultimate, objectives, is to stimulate interest in development of gene therapies for these very rare disorders.
Thank you. To the microphone, please.
Thank you for taking my question. Actually, I have a question regarding the BGTC approach moving forward. I fully understand what you are looking at are gene therapies based on AAVs, but my question is if there's a disease that understanding leads toward, instead of having a gene therapy, designing a molecule, a small molecule, is that also part of the charter of the BGTC, or it moves to someone else, maybe at CBER with a similar charter? I don't know if there's opportunities there.
Yeah, no, thank you very much for the question. I didn't really have a chance to get into the details of what the BGTC is focusing on. So the BGTC is focusing just on AAV gene therapies, and that was selected for various reasons, but primarily because there's considerable experience, in the field with AAV gene therapy. So that was a reason for that. And again, focusing on very rare diseases. Now, what I didn't have an opportunity to touch on, but I'll take the opportunity now to do is, what does success look like for the BGTC? And success isn't necessarily that all of these products that are part of the pilot will result in therapeutic products that show benefit. That would, of course, be the utopic situation, right? The best case scenario. But the BGTC is really trying to look at whether this approach of streamlining, would work for development of gene therapies for rare diseases. So even if some of these programs that were selected for the pilot, even if those products don't show clinical benefit, that's why we do the studies. That's why we do the research. But that doesn't mean that the BGTC is not successful. The BGTC will be successful if we can demonstrate that this collaborative approach, the streamlining of requirements, the regulatory, flexibility, does that, make development go faster, and does that result in, products that would reach patients sooner. But to answer your question, it's just focused on AAV gene therapies.
There's nothing that you look now on CBER side, looking for similar approaches for small molecule drug development as well?
I am not familiar with something exactly analogous to the BGTC, but are you aware of anything?
It would be very interesting to get that talk moving forward. I see Janssen is there, and in the BGTC, I used to be Janssen's employee, and I think that would be a kind of interesting, because there are many metabolic diseases that are gene dependent, and there might be options on small molecules as well.
Okay, thank you. Can we take the next online question, please?
Sure. This is for Brian. What are the recommendations to guide RNA priority analysis?
I'm sorry, I didn't quite hear that. What was the question again?
What are our recommendations for purity analysis of guide RNAs?
Yes, purity analysis. So guide RNA purity really is a direct sort of measure of safety, right? And this go back to the off-target analysis. So if the sequence of the guide RNA is incorrect, that could result in unknown off-targets. So we recommend a threshold of 80% purity for guide RNAs, genome editing components. And we understand that there are actually multiple different sort of methods you could use to analyze guide RNA purity. And so the resolution of the method, your particular method, may vary depending, from other methods that you might use. If for some reason you feel that you cannot meet the sort of 80% threshold, especially for early phase clinical studies, you can provide justifications for why that product would still be safe or effective in clinical trials. However, we still do recommend that you try to improve the purity of your guide RNA through product development in early phase clinical studies so that you can meet that threshold by the time you get to late phase clinical trials.
Thank you. Can we take another online question?
Sure. This is another question for Brian. Would you advise developers of cell-based genome edit products to provide critical component information in the control materials section 3.2 as 2.3 and or as complete 3.2s? And which critical component type would you suggest in providing in which format?
Yeah, so this is advice we provide fairly commonly. And because of the amount of information that we recommend you submit for genome editing components, and it is a fair amount of information, we do not recommend that you submit that in control of materials. We actually recommend that you submit each genome editing component, so the guide RNA, the nuclease, the donor DNA, for example, in individual drug substance sections. They're more ethical to submitting all the information necessary to establish the safety and quality of those components in an IND submission.
Thank you. If there's anyone in the room who has any additional questions, please feel free to come up. But let's take another online question.
Okay, so this next question is for Gopa. Gene therapies are expensive. The affordability issue is a challenge even in high-income countries. What will it take to make gene therapies a reality in low and middle-income countries?
Yes, that's a question that does come up. The promise of these therapies, I think, is indisputable. But whether or not it's a practical reality in low and middle income or resource limited settings is a very valid question. So I guess I'll tackle it from two different perspectives. On the issue of affordability and the cost of these gene therapies, with advances in technologies and also with increasing focus on gene editing, it is anticipated that the cost will come down, between competition, advanced technologies, and use of gene editing. It's anticipated that the cost will come down. So that's the cost part of it. But the other reality in low and middle income countries is, as I mentioned, the vast majority of these countries don't have a regulatory framework. They don't know what these products are. They know they're coming into their countries and they know they need to be regulated, but they don't have the experience, they don't have the knowledge, and they don't have the regulatory framework to do that. What we do know is that there is a lot of development going on for gene therapies that are targeted for Africa, for example, for sickle cell disease or for HIV. So clinical trials are going to be taking place in those countries. And one of the reasons that we've been proactive in working with the WHO is to really start to sensitize these countries to know that these products are coming. Because as you know, we've seen it with the COVID-19 vaccine experience in the US. If people aren't familiar with something, they're not going to be wanting to take it. So the idea right now is to start working with low and middle income countries to start the education process about, what these therapies are, and start to get some things in place, not just a regulatory framework, but of course, looking at monitoring of patients, is something that is really important to do. So the dialogue is starting now because the anticipation is that there are going to be clinical trials in Africa, for example, 5 years down the road. So to answer the question of is it a reality, it's not today. But the important thing to do is to start preparing now for down the road when it could be a reality.
Thank you.
So this question is for Brian. So when you think about gene editing products, a lot of times they are specific to a mutation. And when you want to treat patients of a disease who may have different mutations, it can get challenging to think about the CMC requirements for each specific gene editor. So how do you incorporate, or what suggestions might you have for sponsors when you think about things that could be leveraged? I see the platform guidance that was released yesterday that talks a lot about cross-referencing and master files and things like that. But if you're, a lot of these products are a little bit earlier in development. So what sort of strategies exist, especially around stability and things like that, to really facilitate expedited development for rare diseases with genome editing products?
That's sort of a good question. Certainly, it depends on sort of what level of platform we're talking about. What might be a simple start to that would be using the same editor with swapping out a guide RNA for repair of different mutations in monogenic diseases. That's something that we're seeing now at the agency. People are sort of being able to leverage some data, whether in preclinical studies or sort of in manufacturing CMC assessments. So that's something that I think the agency is discussing internally, and something that we'd like to be able to provide more advice on in the future. But at this point, we don't have a lot to offer, other than a few things, like I say, swapping out a guide RNA or things like that. But
Okay, I think we can take another question at the microphone.
Hello, I have a question, please. I'm just curious, since we are talking about the rare diseases, like, is there any minimum number for patients that can be participating for a clinical trial for those diseases? Like, if there is a very rare disease was newly discovered and only a few patients, like 3-4 patients, for example, can a clinical trial be admitted or accepted based on this number?
Yeah, so we certainly have IND submissions that, it's a patient trial N of 1, right? Where there may be one patient that has a particular disease or mutation that the product is intended to treat. I think generally sponsors look to sort of group maybe small different patients together that have very similar diseases. Maybe they have slightly different mutations. When we're talking about genome editing products, but certainly, yes, there are INDs for as little as one patient.
Okay, thank you.
Okay, I think we have time for maybe 1 or 2 more online questions.
All right, this question goes to Brian. Given that the limited samples can be generated practically prior to IND, can you hear me now? All right, we're going to start that over again. This is given that samples can be generated prior to IND, would N equals 3 donors be considered as sufficient for GE off-target assessment in non-clinical studies for initial IND?
So for confirmation of an target analysis, it's really important that the particular off-target you're looking for is actually present in the donors that you're screening. So So if you have a list of potential off targets, 3 donors may or may not actually contain every off target on that list. And so it's important that you sort of select a list that you're essentially testing all the off targets on the list. So specifying a particular number of donors would not be easy in that case. It really depends on the frequency of the off target you're looking for in the patient population. And so it may require that you do analysis on more than 3 donors. So that's the important aspect, is that you're testing all of the potential off targets.
Okay, thank you. Do we have one more online question?
Okay, so this question is for Gopa. You mentioned the availability of publicly shared IND submissions and official FDA communications. Where can we find this?
So what I mentioned is that, NIH is the sponsor for 5 of the clinical trials that will be part of the BGTC, has made the commitment to share this information when it's available. So it's not there yet because the trials have not started yet, but that's a commitment that the NIH made. So they plan to publicly post, the IND submissions to FDA, any communications that may take place between the sponsor and FDA in terms of interact or pre-IND meetings. Their plan is to post all that publicly so that it's available to others. But it's early in the process, and these trials have not started yet. In fact, INDs have not, INDs are still pending. It's still a work in progress. So short answer is it's not there yet, but that's the intent once the trials are authorized.
Thank you. So I think that concludes our session today. I'd like to thank the audience for your attention and for the excellent questions. And I'd especially like to thank Gopa and Brian for wonderful presentations and for handling the questions and keeping strictly to time. So can you join with me in thanking our speakers? Thank you. We'll pick up after lunch. since 1992 and FDA since 1990. Prior to joining FDA, Lorrie worked as a public health advisor for CDC in Atlanta, Baltimore. She holds a bachelor's degree in industrial relations from the University of North Carolina at Chapel Hill. And her talk today is an overview of CBER's Office of Communication, Outreach and Development. Please join me in welcoming Lorrie for her presentation.
Good afternoon, everyone. As Graham said, my name is Lorrie McNeill. I'm the director of OCOD for short. I'll be using lots of acronyms this afternoon, so I apologize in advance for that. For today's learning objectives, move ahead here. I will describe OCOD's roles and responsibilities, provide an overview of the office's commitment to communication and transparency, and highlight the office's contributions to CBER's overall public health mission. So who is OCOD? OCOD is one of 8 offices in CBER. We are comprised of 3 divisions, including the immediate office of the director. I'll spend some time describing the work of each of these divisions this afternoon. The divisions are the Division of Communication and Consumer Affairs, the Division of Disclosure and Oversight Management, and the Division of Manufacturers Assistance and Training. First, we'll start with DCCA for short. DCCA is comprised of 2 branches, the Consumer Affairs Branch and the Communication Technology Branch. The Consumer Affairs Branch, or CAB for short, has a wide range of responsibilities with regard to our stakeholder interactions. CAB handles all calls, emails, and written correspondence from consumers, patients, and healthcare providers. Calls may come into CBER via the agency's 800 line, CBER's direct toll-free 800 number, or from other centers and offices in the agency. CAB also works closely with the Office of the Commissioner on correspondence that may be directed to the Commissioner or other senior agency officials regarding CBER-regulated products. Prior to the COVID-19 pandemic, CAB handled between 8,000 and 10,000 inquiries each year. Along with the office's Manufacturers Assistance and Technical Training Branch, the office handled about 15,000 a year. But to give you a sense of the pandemic's impact on our work, this number ballooned to 18,000 just for consumers and healthcare providers during the public health emergency alone. Oops, one too far. One subcategory that's included here are complaints. CAB, along again with the Manufacturers Assistance and Technical Training Branch, handles complaints from consumers, healthcare providers, and others about CBER-regulated products. This may include approved products or unapproved products, including those that are being studied in clinical trials. OCOD staff will collect information from the stakeholder and share with the appropriate staff in the center, including the Office of Compliance and Biologics Quality, to determine if further follow-up is warranted. There we go. CAB is also the lead for handling media inquiries. The branch works closely with FDA's Office of Media Affairs to develop responses, draft press releases, and other proactive communications, and is responsible for consumer and healthcare professional outreach. CAB also works closely with the Manufacturers Assistance and Technical Training Branch, in another division, on the center's exhibit program. CBER traditionally has exhibited at scientific meetings and conferences sponsored by healthcare, professional, and industry trade organizations. Need to learn how to use my clicker. Finally, the branch handles a number of special communications projects on behalf of the center, such as creating content for the center's website, and working on information and projects that address misinformation and disinformation, particularly in recent years related to vaccines. The other branch in DCCA is our Communication Technology branch. This is the branch responsible for our electronic communications, most notably the content development, design, and management of CBER's website. Branch staff team up with staff across the center to develop content for the site, including design, graphics, etc. For example, we recently published redesigned pages on CBER's research program, which feature information on the center's principal investigators and ongoing research projects. CTB is also responsible for the center's listserv and social media accounts and works closely with the center staff and the Office of the Director to develop messaging around product approvals, workshops and seminars, policy decisions, and scientific publications. Finally, CTB is the CBER point of contact to the agency's internet policy groups and web governance council. Our staffs are experts in ensuring our pages are 508 compliant, and they work collaboratively with agency staff on navigation and user experience. The next division in OCOD is our Division of Disclosure and Oversight Management, or DDOM. DDOM is the largest of the three divisions in the office and is comprised of 3 branches and 2 special disclosure units: the Congressional and Oversight Branch, the Access Litigation and Freedom of Information Branch, the Electronic Disclosure Branch, and as mentioned, the 2 new disclosure units. Starting with the Congressional and Oversight Branch, this group coordinates responses to congressional inquiries and requests from members of Congress, including correspondence, briefings, and document requests and hearings. OCOD's responsibility is for legislative members of Congress, and we work closely with CBER's Office of Management, which handles inquiries from appropriators. COB also manages GAO inquiries and audits on behalf of the center, and OIG studies and investigations. The next branch is the Access Litigation and Freedom of Information Branch, whose name gives away exactly what they do. The branch responds to FOIA requests related to CBER's regulatory portfolio. The branch also leads the center's FOI litigation response. CBER is currently involved in FOI litigation in which the same court in 2 separate lawsuits has ordered the center to produce a record number of pages per month. In the ongoing litigation, that is 180,000 pages per month, which far outpaces any court-ordered production that we've ever been aware of. We recently completed the first litigation in which we produced 1.5 million pages of records at an average rate of over 50,000 pages a month, which until more recent litigation was far more than any other case in the US. Excuse me. Both of these cases involved records for the COVID-19 vaccines. In addition to producing records, the branch prepares declarations for ongoing FOIA litigation and makes disclosure determinations for documents used to support FDA in court proceedings beyond FOI litigation. The next branch is the Electronic Disclosure Branch, or EDB. This branch is responsible for proactive disclosure activities. This includes disclosure analysis for documents that are posted on the website, either as required by law, such as the FDA Amendments Act and the electronic FOIA amendments, as well as documents posted per center policy. EDB also performs disclosure analyses for international information exchanges that are conducted under MOU with our international regulatory colleagues. Additional responsibilities in the branch include conducting disclosure analysis of documents for advisory committee meetings, prior to web posting and prior to being transmitted to the advisory committee members, and performing disclosure analysis consults for other agency components that may involve records where CBER regulated products are included. Finally, as you might imagine, EDB is also providing assistance with our ongoing litigation as needed. This brings me to our two newest units, the special disclosure units, which were created last year. These were also put in place to help with the overflow of disclosure work in the division. Most notably, the current focus, again, as you might imagine, is our FOI litigation. The third division in OCOD is our Division of Manufacturers Assistance and Training. DMAT is comprised of 2 branches and 1 staff, the Career Development and Directed Training branch, the Technology Training staff, and the Manufacturers Assistance and Technical Training branch, which I've mentioned previously. Oops, sorry. The Career Development branch, which we also refer to as CBER Training, is responsible for the center's career development programs, which include the development of core and specific job competencies for staff in specific job series, as well as overseeing CBER's professional development program. We also have a leadership development program for non-supervisors and coaching for managers, as well as team development opportunities, which are sessions that may be used for team building within work units across the center. I'll catch up to myself here in a minute. CBER Training also maintains IACET accreditation, which allows us to offer CEs for the courses that we offer in our curriculum. Overall, over the last calendar year, we've offered more than 120 courses and seminar series for CBER staff. The technical training staff works closely with CBER Training to develop online courses for the center. This was crucial during the public health emergency and allowed us to continue to offer critical courses such as new reviewer training and biologics law while staff worked in a remote posture. The group also developed an online tool for employees and supervisors to put together individual development plans. IDPs are used to assist employees in career and personal development to help them reach short and long-term career goals. Finally, TTS works with offices across CBER to develop visual content, not only for training purposes, but also to support outreach and educational efforts for external audiences. The third group in DMAT, and I will apologize to my folks in the room for getting this wrong on my slide, is the Manufacturers Assistance and Technical Training branch. The branch is the lead for inquiries from regulated industry and individual sponsors who may have questions or need help navigating CBER. As I mentioned previously, along with the Consumer Affairs branch, the 2 branches generally handle approximately 15,000 inquiries per year. And the questions we receive most often are how to submit an IND, including expanded access and emergency INDs, importation of CBER regulated products, and 351 versus 361 HCTPs. MAT-B also collaborates with the product offices in CBER to coordinate industry outreach and workshops such as this. And they cover a number of topics, including things like proposed and new guidance. The branch also serves as the point of contact for liaison meetings with industry trade organizations. We've seen a steady increase in requests for these types of meetings from industry organizations in recent years. They are generally held annually and take place online, virtually, and are intended to cover topics of mutual interest between the center and the industry organization. Finally, MAT-B handles all speaker requests for CBER participation in external workshops and conferences. So now it's time for the ever popular challenge questions. Let's see if you paid attention. OCOD handles how many stakeholder inquiries each year? A, 5,000, B, 1,000, C, 15,000, D, 20,000. By show of hands, A, B. Right answer would be C. I think I said it about 6 times. Challenge question number 2. Which of the following statements is not true? OCOD staff perform lot release testing. OCOD staff perform disclosure analyses. OCOD staff participate in the complaint response process, or OCOD is accredited by IACEP or CEUs. And if you said A, you would be right. Finally, I've included contact information for our office and for the center in case you have inquiries. For industry inquiries, the address is fairly straightforward. It's industry.biologics. For consumers, it is the acronym of our office. And I've also included the URL for our website. In summary, I hope I've given you a glimpse into our commitment to providing outstanding customer service to our stakeholders, as well as transparency in communicating about CBER-regulated products. We are committed to ensuring that all of our communications support CBER's public health mission by providing clear, concise, and factual information. And I look forward to taking your questions. Thanks very much for having me.
Thank you, Lorrie. That was a very good presentation. We look forward to a question and answer session. I'd like to introduce our next speaker, Scott Brubaker, who is the Director of a Division of Human Tissues in the Office of Cellular Therapy and Human Tissue in the Office of Therapeutic Products in CBER. Prior to his joining us in October 2016 as the Director of the Division of Human Tissues, Scott spent 12 years as Senior Vice President of Policy at the American Association of Tissue Banks, where his duties included the development and management of association policies, professional standards, and guidance documents, and oversight of the accreditation program. Prior to that, Scott acquired 18 years of practical experience involving organ donation and tissue banking, while holding various management positions as an organ procurement organization and tissue bank in Virginia. Scott has authored or co-authored more than 40 publications and is co-editor of 3 essential guides for tissues and cells that cover donation, processing, and clinical use. His talk this afternoon is HCTP donor screening testing and requests for exemption. And let's look forward to a very interesting presentation. Scott.
Thank you. And thank you, Lorrie, for giving me a little bit more time so I can digress like I usually do. Okay, so I'm happy to be here to just give you an overview, really, of screening and testing for donors of human cells, tissues, and cellular and tissue-based products. And part of the regulations that allow you to request an exemption from the regulation. So we'll go over that because that's seen with some of the gene and cell therapy products that we review. I do like to include this informal communication disclaimer because my comments are an informal communication and represent my own best judgment. These comments do not bind or obligate FDA. I'll be using terms that aren't really FDA terms sometimes, and I'll be trying to relate some of my experience working in the field before I came to FDA. And I like to put this here too in presentations given anywhere, because when the speaker doesn't have financial interests, then you don't get that bias that can creep into a presentation when it's not this way. So I just thought I would put that there too. As you know, the mission of the FDA is to protect public health. But I just want to make sure you realize that if you're a manufacturer of a product, it's for a certain therapy, of course, and it's going to treat and hopefully cure possibly a disease. It's your mantra too, is to protect public health. And we're going to go over a couple of these issues and actually history of transmission of disease in cells and tissues and medical products of human origin. So just think about that and how it can affect your evaluation of the safety profile and that characteristic of your product. So hopefully at the end, you'll understand the medical products of human origin have transmitted a wide variety of disease agents and diseases. You'll be able to describe, hopefully, just in general, requirements for HCTP donor screening, donor testing, and making a donor eligibility determination. And then also, I hope you'll be able to identify when a request for an exemption or alternative is indicated, how to submit it, and helpful tips to consider when you submit it. So this isn't an all-encompassing presentation, but you'll see that history can teach us a lot. And you see that it's constantly evolving. So we've had transmissions happen in modern history that are pretty surprising, and we need to be aware of that. So over the next, I think, 6 slides, you'll see some adaptations into tables of information from a chapter in a book. Now, the book was published in 2012, so it covers history back to 1953, 54, up to 2011. And this slide only shows transmission of disease by quote unquote tissue allografts, conventional tissues, bone, tendon, cornea, vein, heart valve, nerve, skin, those types of tissue grafts. And you can see that when you look at this in just a general way, that the highest risk is with fresh or refrigerated, frozen, and cryopreserved tissues. Makes sense. But once you see it on a table, it really makes sense. So when you look at this too, when you look at data, there's usually an outlier. And the outlier here is freeze-dried dura mater. That transmitted Creutzfeldt-Jakob disease to a little over 100 patients or recipients, if I remember correctly. It was a long time ago, but it's possible. And that was due to pooling of many dura from dura from many donors together in the manufacturing process. If you've never read about that, you should do that. Now, freeze-dried dura also transmitted tuberculosis. But now, when you look, I put red font at tuberculosis, because I think the last time there was a transmission that was included in this chapter was around 1953, 1954 in the UK. But we experienced in the United States, this 2021 and 2023, 2 outbreaks from a bone matrix graft. It was frozen. And it caused, more mortality. And significant morbidity. But the other thing that was really interesting about tuberculosis, healthcare workers taking care of the recipients who were infected also got infected. So when you're thinking about all of this, it's not just about the recipient. In modern times too, bacteria, there could have been a cryopreserved placental-derived graft that was investigated by CDC that transmitted disease. Can't remember the name of the bacteria offhand. So if we look at tissue and line it up with blood, organ, and hematopoietic progenitor stem cells, and look at, those are different types of medical products of human origin, you can see the disease and disease agents. It's a set of usual, as you might call them, usual viruses and bacteria. Those are the ones that are common across all of those types of MPHO. Now it's less common across all the types when you look at specific viruses like dengue, West Nile, and rabies. Tuberculosis is there, but it hasn't been known to be transmitted by blood or hematopoietic stem cells. And then you see parasites come into the picture as well in history. So we're not done yet, there's more. tick-borne diseases by blood, a non-tuberculous mycobacteria from a refrigerated arterial graft for tissue, and organs. Organs are interesting too, because they're like a human papillomavirus transmission occurred from a hand transplant. And a hand transplant in the United States is considered an organ. It's a vascularized composite allograft. So it meets the definition of an organ because they revised that definition along the way. But you can see that some of these, of course, you can't really test for. And can you even screen for it? Maybe not. It just depends on the donor case, the donor that's used for these cases. And hematopoietic stem cells, you can see scrub typhus. But another point to be made is that these are all refrigerated NPHOs. So today there's disease transmission that's been attributed to assisted reproductive technology procedures. I'm not going to go over these in detail, but you can see that semen and ovum and embryo, all infections. There can also be metabolic and genetic conditions that are transferred, and even an antibody-mediated conditions from an effect from the gestational carrier to the newborn. There's HDN, hemolytic disease of the newborn, neonatal alloimmune thrombocytopenia. And if I can quickly, there's a story. My wife and I know a couple who sought ART for family planning, had a gestational carrier. When the baby was born, within a day or two, it was recognized they had GALT, gestational alloimmune liver disease. GALT, sorry. They recognized it quickly, and the baby is fine today. The therapies available today really saved their life. But it can happen. Neoplastic diseases, of course, too, can be transmitted. So here's a list. But again, I just wanted to show this to you so you can see the relationship again to refrigerated products, of medical products of human origin. So let's get into the regulations, the all ever exciting regulations for donor eligibility. The selections, again, are limited, and I'm not going to go into a lot of detail. So here's our regulations. It's divided into 6 subparts. There are final rules for most of the subparts for B, C, and D and E, and you can see that what they cover, and we'll be focusing on donor eligibility, but go into a little bit about current good tissue practice requirements as well. So I've been talking about these HCTPs. They mean articles containing or consisting of human cells or tissues that are intended for implantation, transplantation, infusion, or transfer into a human recipient. So it encompasses a wide variety. You know, our stakeholders are diverse. They don't even really, the groups, professional groups, don't talk to each other a whole lot. They don't really interact. So it's really interesting that you have to understand the HCTPs do come from living donors or deceased donors. So here's just some examples and pictures. HPC apheresis on the left in the back, and the bag there. And that's refrigerated. The cornea is refrigerated. Then you have frozen bone and cartilage, or just frozen bone. That transmitted disease 20 years ago, and the recipient died of a Clostridium infection. It was just a small plug from one of those grafts. But that's a femoral condyles that are cut in half. And then you see, and that's just simply frozen. It's in a freezer. It's not control rate freezing, nothing. Then you have a pulmonary heart valve that's cryopreserved. A small patch from one of those PAs, like the right PA that's there. That transmitted hepatitis C. It was listed on the other slide. Just a little patch, 2 by 2 centimeters. And then you see semen stored in liquid nitrogen dewar in the vapor phase or in the liquid nitrogen. And you know that can transmit disease. And then you have freeze-dried or lyophilized allografts, such as bone. Bone is the most common that's transplanted today. That's corticocancellus, a picture at the top, and the bottom one is demineralized bone matrix. The most popular, shouldn't say popular, the most, the highest number of allografts transplanted today are probably demineralized bone or have a demineralized bone component. I received a cancellous graft. How many in here have received a tissue transplant? Yeah, there's two of us, three maybe. Yeah, oral, dental surgery is really common where they'll use some bone to rebuild your alveolar ridge to implant the post for a tooth. Okay, so one of the other things I'm not going to get into detail is with the tiered risk-based approach that we've taken, that FDA has taken to the regulations and then for governing HCTPs. But there are 4 criteria in 1271-10A. I'm not going to go into those: minimum manipulation, homologous use, combined with another article. Does it have, whether it has a systemic effect or is dependent upon the metabolic activity of living cells. I'm not going to go into that. That's not this presentation. That would be a separate one. But HCTPs that meet all the criteria in 1271-10A can be regulated solely under Section 361 of the Public Health Service Act and these regulations that we're going over, and pre-market review is not required. If you fail any one of those criteria when you look at your product, then it's regulated as a drug, device, or biological product under the Federal Food, Drug, and Cosmetic Act or Section 351 of the PHS Act and applicable regs. So the regs in 1271, Part 1271 still apply. Donor eligibility still applies, and certain CGTPs apply. So there's parts of it that you still do apply. So pre-market review, of course, is required, IND, BLA, and so forth. I think we call these 361 HCTPs for the first one and 351 HCTPs for the second one. Oh, I'm sorry. It's applicable, the rule for donor eligibility and really the rules, tissue rules, are effective from HCTPs recovered on or after May 25, 2005. There was a 1270 rule that was in place since 1997, and then in 2022 that was revoked. And that's another talk too. Our regulatory authority is here under Section 361 of the PHS Act. Because we're an agency of HHS, the Surgeon General and the Secretary of HHS delegated this authority to FDA to create and enforce regulations it deems necessary to prevent the introduction, transmission, or spread of communicable diseases from foreign countries into the U.S. or from state to state. So because HCTPs contain components from the human body, they do pose risk of carrying pathogens that could cause disease in. And this is interesting. I don't know if you've noticed this before, but it says in healthcare personnel, other handlers of tissue, there's recipients that everybody thinks of, and family members or other close contacts of recipients. So that TB, what happened with TB is a case where you saw, and I don't think it's been investigated if family members have also become infected of those healthcare workers that were infected. Quickly, I just wanted to show a couple of definitions. I'm going to mention these later on other slides, but there are 40 definitions in the regulations in 1271.3. There's the definition for manufacture. We're going to focus on donor screening and testing of the cellular tissue donor. And then plasma dilution, the sample that's used for testing has to be qualified in a couple of different ways. And one of them is to look at whether infusions or transfusions have could affect the test results run on that sample. And then a responsible person, I'll mention a couple of times because that's the person who's authorized to perform designated functions for which he or she is trained and qualified. And that directly relates to donor eligibility determination that has to be documented by a responsible person. So in the very beginning of this subpart, It does set out requirements for determining donor screening, determining eligibility using donor screening and testing, but it's also considered that subpart C is a part of subpart D. It's part of good tissue practice. So again, a donor eligibility determination is required. There are some exceptions, and it says 127190 is listed there, and that includes exceptions for autologous use and for reproductive use. And then there's a prohibition on use. You must not, HCTPs must not be implanted, transplanted, infused, or transferred until the donor has been determined to be eligible. There are some exceptions for that as well, but it doesn't really apply. Urgent medical need doesn't really apply to 351 products. And then there are some other exceptions in 127165B. So as we go further into the subpart, there's sections that cover procedures that you have to establish and maintain. That determination of donor eligibility is based upon screening and testing. The donor has to be eligible, and it's, again, documented by a responsible person. And then there's a provision for accompanying records. And you can see there's a distinct ID code that's required. So you have to establish that. There's summary of records and other things there, and record retention requirements, which I think I do get into a little bit here. There's a section for quarantine before the donor eligibility determination is completed. There's a section for storage from a donor determined to be ineligible, and when an ineligible donor's tissues or product can be used, when it's not prohibited, when the donor is ineligible, there's also a section that describes that. Of course, you screen. How do you screen a donor? It's looking for risk factors for and clinical evidence of RCDADs, as well as communicable disease risks associated with xenotransplantation. You know, xenotransplantation doesn't, the risks associated with that don't fit as a disease agent or a disease, but it's part of the requirements for screening. And relevant medical records are described there. And here's just a graphic that shows what relevant medical records can entail. A current donor medical history interview, a current report of the physical assessment or physical examination. If the physical exam is for a cadaveric donor. Other records that are, if available, such as additional lab test results, medical records for deceased donor, coroner and autopsy reports, and other relevant information from other sources that you might obtain. One note I want to make here is that for a donor medical history interview, the blood sector, I call them sectors, they have a form that they use that's FDA has reviewed and includes in guidance as a good form to use. There is nothing like that in the HCTP sectors. There is one form that OPOSI banks and tissue banks tend to use, but they're not required to use it. And we have all these other sectors, reproductive and et cetera, and they don't really use one form. So we look at many different types of forms when we look at records for a donor to evaluate whether the donor eligibility criteria are met. Now in this subpart, of course, we just had donor screening, now there's donor testing. And I'm going to actually go into those in more detail, so I'm not going to talk about them right now. We also have requirements for screening and or testing if the product is viable and leukocyte rich, because the cell-associated diseases are then implied because that's a risk now. They can transmit disease if viable leukocytes are present. And then there are other exceptions here for labeling. You know, when there's an ineligible donor that can be used, then you rely on labeling and prominent warnings and labeling that the physician or the clinician who's going to use the product will then discuss with the recipient So when requirements aren't met and you submit an exemption, a request for exemption or alternative, if it gets approved, it may have definitely, probably will affect the labeling that you'll have to use. So let's go a little bit into qualification of the specimen. I should say control of the specimen, the blood specimen from the donor, or whatever specimens are used. But they're obtained or collected at the time of recovery, or it's allowed up to 7 days before or after recovery. So you want to have a current situation for testing. Now, for donors of peripheral blood stem progenitor cells, bone marrow, if it's not accepted in the regulations for 1271, or oocytes, the specimen for testing may be collected up to 30 days before recovery, and the 7 days after time period still applies. Now, that's done because those donor types or the recipients of those products may undergo conditioning regimens beginning more than 7 days before recovery. And this is all found in 1271 ADB. So it permits collecting the donor specimen for testing up to 30 days before recovery in those scenarios only. So we've received questions about, does that apply to my semen donor? Does that apply to my PBMC donor or my cord blood donor? And the answer is no. There's no conditioning regimens involved in that for either the donor or the recipient. So plasma dilution, that has been related to transmission of HIV. It was an organ donor back in the 1980s, but it was realized back then that this can happen. So a donor is ineligible if plasma dilution is sufficient to affect the results of communicable disease testing, unless you find a sample that's pre-transfusion, pre-infusion, which can happen in an emergency room situation. I know this is more about deceased donors probably, but it applies to living donors. If you have a living donor who's you're getting the cord blood, I should say the cord tissue from, or the placenta from, and you're making a product, they could have had an event at delivery that they got infused, transfused, and if you collect a sample that's after those events, those administrations, you would have to qualify that sample, and it may not qualify because of plasma dilution. So don't think this is only applicable to deceased donors, is my point. You can apply an algorithm in those cases. It's basically, there's an example in the guidance document for donor eligibility from 2007. A lot of folks do follow that as it's written, and it allows for up to a 50% dilution. And that should not affect testing. But just understand the testing today too, for serologies, the test is run on a dilution of the sample. So if you dilute it even more, you have more of a chance, of course, of getting a false negative. So now we've looked at the timing of the specimen collection. We've looked at plasma dilution. So that's the controls for the specimen used for testing. But now we have also controls for the laboratory. You want to have a high level of confidence that the laboratory is going to perform the test correctly, because humans are involved, so there can be mistakes along the way. But that's really why the CLIA certification requirement is here, or CMS equivalent. It's to really make sure that they're following the manufacturer's instructions. They're not creating their own instructions or validating the test themselves, because as you'll see, the test kits themselves are already licensed, cleared, approved by FDA. So the instructions must be followed. So that's a check in your checkbox for your safety profile for your product. So I've touched on RCDADs already, used that term. It's relevant communicable disease agents and diseases. They're listed in the regulations at the time that the regulations were promulgated. There was a set, but with their new RCDADs can come along. And they're notified, we notify industry through guidance documents for that. And some of the definitional elements or factors that are considered when there might be a new RCDAD is the risk of transmission by an HCTP, and includes known transmissions and the potential for transmissions. But included in this is also the prevalence or incidence in the HCTP donor population. That's one of the factors within that one for risk of transmission. The severity of effect, it poses a significant health risk, morbidity or mortality, and availability of appropriate donor screening measures and tests. We don't always have a test, but you can always screen. So here's the list. Back in when the regulations were published in 2004, it was HIV 1 and 2, HBV, HCV, human transmissible spongiform encephalopathy, which includes, of course, CJD and variant CJD, and Treponema pallidum, the agent that causes syphilis. And then when the guidance document came out for donor eligibility in 2007, 3 more were added: vaccinia virus, sepsis, and West Nile virus. And in 2016, separately, the Zika guidance came out because that became a public health issue for sure. And it was actually that guidance that identified it as an RCDAD was just withdrawn Monday a week ago. So it was no longer considered to be an RCDAD because of the prevalence of incidents in the United States and basically worldwide. I was going to try to draw a line through Zika and then date and initial it, but I thought that was too geeky. And then there are other requirements too for the cell-associated diseases, HTLV for viable leukocyte-rich cells and tissues, and for reproductive cells and tissues is chlamydia trachomatis and Neisseria gonorrhea. So this table shows the different agents, if it's required for all HCTPs or just some, whether screening is required and testing. So you'll see TSEs, we don't have any testing for that. So we entirely rely on donor screening to identify risk. Sepsis is the same way. Vaccinia, again, Zika can be removed. We never really had a test that would work for us that you would test from the peripheral blood. Now CMV is not an RCDAD, but viable leukocyte-rich HCTPs can transmit the disease. It's ubiquitous in our population. So what do you do with that? You have a testing requirement and an expectation to share that information with the HCTP when it's distributed. So clinicians can make an informed decision whether that product should be used for their patient. Now there's one, it's not an RCDAD either, but it's not listed here. Which risk is not listed? Xenotransplantation. I have to talk to staff because we use this slide a lot, and I just noticed this like the other day. Xeno's not there, but we have to screen for risks associated with xenotransplantation. So when we talk about FDA licensed, cleared, and approved tests, these are the ones that are adequate and appropriate for reducing risk for each of those disease agents. And just to point out that there can be more than one test needed. So hepatitis B virus, you need an FDA licensed donor screening test for hepatitis B surface antigen, total antibody to hepatitis B core, which includes, of course, IgG and IgM, and a NAT test for hepatitis B. Other ones as well have more than one test that's required to adequately and appropriately reduce risk of transmission. You can see some are FDA cleared, like T. pallidum, it has its own guidance document where it describes the non-treponemal or treponemal testing when it can be used. CMV is an FDA cleared donor screening test. And chlamydia and Neisseria are diagnostic tests because that's really all that's available, but it's been tested in an asymptomatic low prevalence population and then that NAT test is acceptable to use. So you can see there are other guidance documents here for when that West Nile virus NAT was added and also HBV NAT in 2016. So the related guidance documents are here for you, and you can see that if you know about our CBER guidance agenda that was published in January, actually, I think for about a year and a half, 2 years, that first guidance document has been posted as being updated, and it is. We just don't know exactly when it will be published, but we're hoping soon. And you can see over the years, after 2007, it was 2016, there were a couple other guidance documents related to donor testing, donor screening. And then we had a removal of a risk factor, HDCFCs, or human-derived clotting factor concentrates, was no longer considered a risk for a donor of HCTPs. And then the Zika guidance, which has been withdrawn. If you want to learn more about the tests that can be used, you can go to our website that we host and OTP, testing HCP donors. That's the one to go to. You can also use the one by OBRR if you have a living donor, but you have to be careful what you find there and how you interpret it. Knowledge check. Which of the following disease agents is not an RCDAD? Correct. Cytomegalovirus. So now to go into the last part of the presentation, I'm going to cover a part of the regulation in 1271-155 that allows for exemptions and alternatives to be submitted. So you have your product. Maybe it was recovered and half of the screening was done, or was done by folks in another country. That's where the donor is, that sort of thing. You have to evaluate all of that. We will do it as well, but you should know it really well yourself. So you can request an exemption or alternative to any requirement in subpart C or D, so donor eligibility or current good tissue practice. You submit it to our super office director and include information that justifies the requested exemption from the requirement, or a description of a proposed alternative method of meeting the requirement. And you can't start using this request until it's approved or granted, as we say. So what if requirements are not met? Go to our webpage, look at what needs to be submitted, but I'm going to offer some helpful hints too here next. But you have to convince us that it's okay that some of these things were not done. This graph was a little funny. I found a mistake on it too late. It missed 5 other people missed this mistake, but you can see that we entertain exemption requests of around 50 a year for the past 2 years, and most of these are reproductive HCTPs. But some of them are 351 products as well. So what do we see most often as non-compliance? Required donor screening, the donor medical history interview form, which there is no standard one people follow, mistakes are made. Required donor testing, specific tests were not run. That qualified certified laboratory was not certified by CLIA or a CMS equivalent. The donor eligibility determination was either done incorrectly or was done based on incomplete information. And then we also get, of course, requests for pooling. We've seen this can happen in many scenarios, but one of them is when the human cells or tissues are originally recovered for research. Then they become a component of a product intended for clinical use in human recipients. And that's where gaps are found. Up front, the complete profile was not taken. This guidance document is important for you. It describes core CGTPs, but really for you, there's a section, an entire section that's dedicated to 351 products. So let's go into that. So it's in section 3 at E. Do I have to follow CGTP requirements if my HCTPs are also regulated as a biological product or device? The answer is yes. There will be requirements, the CGTP requirements, as well as drug current good manufacturing practice requirements or device quality system regulations requirements apply. Now there are, I have some tables coming up next that well, it's not a table, it's a list of comparing CGTPs to quality system regulations and to CGMPs. And you can see there's a list of almost a dozen of our sections, our requirements that are not covered by the other regulations for drugs, devices, and biological products. So it is important to note that the CGMP and QS regulations supplement the CGTP requirements in this case. And in the event that a regulation in Part 1271 is in conflict with a requirement in Parts 210, 211, or 820, the regulations more specifically applicable to the product in question will supersede the more general. So here is the list, and subpart C is number 1. Some of the other ones, I'm not going to go into those. They do apply, and you do have to show that you've addressed them. They are important, but I didn't want to focus on that today. One that's different between the two lists for CGMP and QS would be audits. It's only in the CGMP list that it's not really covered by any of the CGMP requirements. Then, of course, there's our prohibition on pooling. There's a section on pre-distribution shipment, which you should really look at and follow. And then the other ones are, of course, about packaging and distribution. Record keeping is different. It's for 10 years. And facility cleaning and sanitation records are for 3 years. You know, if you have a viral transmission that can go unrecognized for years. That's one of the reasons that we want to have those records around for 10 years. And the tracking requirements too are very important. So make sure you familiarize yourself with A through G. Helpful tips for submitting a request. In a letter, summarize the information that you have and the regulatory deficiencies you've identified. We might find more. Don't be surprised that we do. The data recovery is very important for applying the regulations and guidance. Your documentation of the donor medical history interview questionnaire. The physical exam, hopefully there was one of your living donor. We want to see the donor test results from the laboratory printout, not your summary of the laboratory printout. I'm sorry, we must do that. Information about the laboratory, trade name of each test, so we can look at those closely. And if you actually do have an FEI number, include that. For helpful tips for submitting a request, if you have a drug, device, or biological product, we encourage that you discuss that with FDA early on, like during an Interact meeting or a pre-IND meeting, so we can start looking at this early on instead of waiting, and then you find out that it's a real big problem. So you should submit the request prior to submitting your application for IND or IDE. We don't look forward to putting something on hold, clinical hold. So we really want to avoid that. But communication's key. Also, what we'll need is a really good description, and normally that happens anyway, but the description of the manufacturing process of your product, description of how you determine donor eligibility and the donor screening and donor testing that was done. And then whether if you do have a mitigating, if you want to mitigate the risk and you want to do in-process or final product testing, please submit what your plan is for that. And that requires robust validation, I would say. I'd like to call it robust. Is your test method suitable for the product? And of course, these are in-house tests. So make sure that's done correctly. Our small division, we do product reviews. You can see INDs were up to 52, actually up to 70 in 2022 and come down a little bit. But BLAs are hanging in there too. So we look at pre-INDs, original submissions, amendments, supplements, and perform inter-center consults, often from CDRH, where we look at HCTP donor eligibility, the applicable GTPs that I just went over. And we also review donor screening tests that have a cadaveric claim. So we have to meet PDUFA and MDUFA guidelines when reviewing those. So we're just about at the end. Really simple question. An HCTP donor eligibility determination is based on the results of number 3. So I've used this slide for a long time. The reason I had the history slides there was that we're not learning from history. We keep repeating history. So I think as you look at your product and its safety profile, you know, look at it closely and see if improvements can be made. Because if you're just at the beginning of product development and you're actually don't have a donor yet that you're using, and you haven't characterized a cell line that you really want to use, moving forward, if you follow all of these regulations and the guidelines, all the recommendations, you're going to be in a good place and you're going to feel much better about it. So I just want to end with some resources. This page, our staff uses every day. I use it every day. It's searchable. The ECFR, it's really good. It's improved over the past couple of years. It used to be kind of not really user-friendly, but it's very user-friendly now. These other pages, you can find the tissue guidances there. Sometimes we'll issue a safety and availability notice for HCTP establishments, so keep a lookout for those. We also have a subscription for CBER updates. It's wise to subscribe, as I like to say. So when guidances are issued, if you're subscribed to this, you're going to get that information. I didn't talk about 127110A very much at all, but we have options for obtaining a regulatory classification for your product. You can come to the tissue reference group or submit a pre-RFD to the Office of Combination Products, and you'll get a recommendation via those routes, or you can obtain a decision from OCP, the Office of Combination Products, if you submit a request for designation. So in summary, use of HCTPs has led to risks to public health due to transmissions of a variety of disease agents and diseases. The DE requirements are applicable to all HCTPs, regardless of how they're regulated. And donor screening and donor testing for RCDADs and screening for risks associated with xenotransplantation are required for making a DE determination. But you do have the option of considering submitting to FDA a request for an exemption or alternative if any DE or CGTP requirements are not met. So I think with that, we don't have the funny picking your brain cartoon up here.
Thank you. Okay, so I'd like to kick off the question and answer session now. Lorrie and Scott are both available to address any questions, and let's go to the microphone.
Hello, and I apologize, Lorrie. My questions are all for Scott. So, the first one is, so one of your slides said that your definition of manufacturer included donor eligibility. Is that true? So does that mean all of donor eligibility needs to be done under a batch record?
It's in donor testing and donor screening. That's
Okay.
part of manufacturing.
Okay.
So then related to that is that you have to make a DE determination. That's a must.
Okay, is it done under like a batch record, or do you, I guess, it's just the documentation that's considered to be?
Those are manufacturing steps. Those are all the steps that define if manufacturing itself. I'm not sure what you mean there.
Okay.
Are you talking about allergenic modified products or cell therapies?
I think it was everything you said that it was applicable to, because I was thinking at the time, because you mentioned plasma dilation, you mentioned a couple of other things. Let's say someone is getting plasmapheresis to collect plasma. When you do the donor eligibility of that, is it just done under the typical documentation, or does it have to actually be a batch record? You know, because if it's manufactured, like for labeling, you would need to do that under a batch record under GMPs.
Yeah, what I would do is just look at donor eligibility and all of those sections that apply and procedures and it's all in there in records, documentation. Yeah, I'm not sure exactly what you mean.
If it's cells but collected for subsequent manufacturing use, yes, they go to the batch record. That information will go to the batch record.
Curious, because you mentioned packaging, the packaging and distribution are exempted from that as well. So you would not have to have those records for packaging and distribution?
If you package, that's a manufacturing function, so then you follow that.
Okay. So a few things that were
Which is in the GTPs.
Right. Thanks.
Okay, next question.
So thank you, but I have a couple of questions for Scott and one question for Lorrie. So for Scott, so I have a question. Didn't we see in for the case for the transplantation from kidney from kidney from pig to human? So do we have any plan to put any testing and screening for different cross species for transplantation?
Didn't hear all of that. I have tinnitus, so I don't catch everything everyone says.
Screening of for xenotransplantation. For how are the animals screened? I believe
For the case from kidney transplantation from the pig into the human, this is done at the hospital in Boston. So do we have any plan for in the future guidance to put in the cross different species in the screening and testing?
Yeah, in the donor eligibility guidance, there are some definitions you'll want to look at. It's what is a xenotransplantation product?
Yeah.
What's a xenotransplant product recipient? And what's a xenotransplantation? There's a definition for that too. So it would include receipt of products from non-human animals that contain live cells, or during the manufacturing process, they were exposed to live cells from a non-human animal. So then now you could be at risk for transmitting diseases we are maybe today even still unaware of, endogenous retroviruses. So, that's the risk. It's I'm not sure if your question was related to it or not, but like a pig valve, or bovine valve, or bovine pericardium receipt of that is not a xenotransplantation product. It's a xenotransplant, and it's regulated, by FDA, but it's not a xenotransplantation product. I hope I answered your question.
Yeah, I think, but yes, you did. And the second one, so in UK that we see that in 1970 to the 1990s, there are a lot of guys with HCV, HIV. So any record in the United States at the time, because HCV was discovered in 1989, HIV was discovered in 1983. So is there any case recorded in the United States for the transmission?
So HIV was discovered in the 1980s.
83, yeah.
83. Was your question whether there are documented human transmissions from tissue before that?
Yes, that's
No, for HIV.
For HIV.
Yeah, I think it was first realized. I have slides I use sometimes. It's too much to put in here. When these events happen, that FDA recognized this is happening, we have to do something, we have to regulate. but it was most, I think, first in semen. Semen, you had HIV and HPV transmissions happen.
And actually, I have a neighbor who did a transplantation in kidney in 1992, and she had HCV at the time, but she still survived.
Okay.
Until now, she's 82 now.
Can we go to the next questioner, please? Thank you.
Well, he's coming up. The regulations of what really stirred FDA to do something, there was an interim final rule in 1993, and they realized that tissue was coming into the U.S. from brokers, South America, Central America, Eastern Europe, and they were trying to sell the tissues to the U.S. unprocessed to U.S. tissue banks. And the U.S. tissue banks reported it to FDA and said, hey, you know this is happening? And that's when they realized they had to put something out to protect the public health.
Lou Bartolo from Senior Regulatory Affairs for the National Marrow Donor Program. So I'm going to ask sort of about the white elephant that's in the room always right now. It's the LDTs. And we're getting questions, I think, more or less regarding some of the testing and so forth for donors. And where should we direct those questions to? Is it really OTP, or should we send them back to CDRH from like network partners? So maybe Lorrie can touch on that.
Well, we have gotten questions, on LDTs, and there, I believe, is a final rule.
Correct.
Right. You can certainly start with us, and if it's something related to screening or testing of donors, then we would work with Scott's group on a response, but it might very well be a CDRH question. And so if it is, then we can make sure we direct you to the right place at CDRH.
Or CMS at both, yeah.
Right, but with the laboratory, some of the testing that they do are not cleared by FDA. They're, the reagents.
No, the situation I think that is being referred to is laboratory developed tests for kind of specialist purposes.
Sort of like if you have an HHV 678 test that's not that someone's requiring or asking about, but it's not actually approved by FDA. So
like the HLA testing and so
correct, because
right.
those are exempt under the rule. So correct. So that's why we're just trying to figure out as we get these questions from across the country, who to really filter them to.
Thank you. We'll go to one more question at the microphone, and then we'll do some online questions.
Hi, Tyler Zimmerman from United Therapeutics. Thank you both for your talk. I had a question about donor pooling. So I guess what would be a reasonable justification for you to see to do donor pooling? And then to follow up on that, if you were to screen all of your incoming donors with the appropriate tests and screening, and then you pool them afterwards, do you still have to do additional testing, or are there any other things that you would need to see in terms of donor pooling?
Pooling's prohibited, so you would have to submit for an exemption, and you'd have to justify why you deem that you must pool. And remember that donor screening and donor testing, these are the minimum requirements, right? And donor testing still has window periods, every test. So if one infected donor's product or tissues or cells are used in that batch, you can adversely affect a lot more recipients. So it's, you have to really justify why you must do that and how many donors must be used. So you've got some work to do to justify all of that.
Okay, thank you.
Okay, let's take an online question.
Okay, this question is for Lorrie. Are the robust training modules and tools for industrial on FDA website?
Adrian, can you turn the microphone on?
Oh. Okay, sorry. All right, can you hear me now? All right, this question is for Lorrie. Are the robust training modules and tools for industrial on FDA website?
Which training tools?
All their robust training.
Robust training.
Are there training tools for available on the FDA
Oh, you mean
website?
the tools that we use to train our staff? No. They're not available on the external website, no.
Can we take another online question?
Yes. Hi, Scott. How are human pancreatic islet cells intended for transplantation regulated by the FDA?
Yes, the allogeneic is regulated by FDA, and so are autologous. But it's a different
I think the question
process.
is like, how are they regulated?
How are they regulated? Yeah, I think I just
351 or 361?
351s for allogeneic and 361 for autologous.
Okay, let's take another question.
All right, this is for Lorrie. What kind of support does your manufacturer assistance group provide to sponsors and manufacturers?
That's a really good question. We are really the entry point for sponsors that perhaps have a new product. Typically, we're not going to get questions from very large companies that are used to navigating FDA. So small sponsors, researchers are often the most frequent inquirers to our manufacturers assistance group. They need to understand the process, who the right contacts are, which office might be regulating their product. So it's really kind of an intro to CBER 101. Would you all agree?
Okay, can we take a question at the microphone now?
Thank you. Question for Scott. So in light of the need for a lot of these CAR T therapies, CGT therapies needing to run global trials to get enough patients, things like that, we're seeing a lot of gaps, as you mentioned, with donor eligibility requirements from regulatory agency to regulatory agency. So my question from your perspective, what do you feel like the exemption is there for? What kind of programs or sponsors is that sort of geared towards in efforts to get these therapies to patients that may have missed this in the beginning?
It's geared towards any product that's going to contain human cells or tissues, whether it's 361 or 351. So I think it was realized early on that can happen. And if you can take mitigating steps that are convincing enough that you've mitigated that risk sufficiently, it can be granted.
Scott, can you just cover one thing? So autologous products, what are the requirements for autologous cell-derived products versus allogeneic? Because the autologous products don't, for example, the CAR T-cell manufacturer, are not necessarily subject to donor
That's true, yeah. Then that's right. I didn't make that distinction, but Graham's correct, yeah.
Great, thank you.
Thank you. Can we take another question at the microphone?
Just for those online, my name is Richard McFarland, Chief Regulatory Officer of the Advanced Regenerative Manufacturing Institute. And Lorrie and Graham and Scott were colleagues once upon a time. I'm going to be kind. I remember what it's like to be there, Scott. Army is really based to make cells, tissues, and organs. So in making complicated tissues and cells, many of our members have the question like, I need to pull from 2 different donors. I get epithelial cells, for this example, from Huvex, but I'm using primary liver, primary kidney lines. So they had to request an exemption. I said, well, request an exemption. How early can they do that? Because it slows them down in product development and it makes their investors nervous. So a little more earlier certainty would kind of move things around. What is the process? And so, for instance, can they submit a request to DHT when they're just got an idea before they talk to IND, even before they interact.
Or interact, so it's really earlier. I think you can. So you don't really even have a review team yet, right?
Right. So they're in a situation where they haven't quite decided what their product is going to be. They're thinking about different cell type. They've got a preferred cell they'd like to do in the pooling. And they need to know that answer to do that preliminary proof of concept data to get to the interact.
That's an interesting question. We need, when we're reviewing an exemption request, though, we're looking at a lot of information that you have. And in that case, you don't really have a lot of information.
But you might have donor eligibility information on the two separate lines and have an idea of what the product might end up like. If you haven't been to the CMC folks yet, and you want to do some concept, and it's like, I don't want to, the investors are telling my members, I don't want to go forward until I know they're going to be able to use that line, these two lines together.
That sounds like something that you might be able to ask in a Type D meeting.
In a what meeting?
Type D.
Type D, okay. That's good. That's a good thought. It really wasn't meant to be a hard question. It was going to be a hard question, but I knew there wasn't going to be an easy answer. Because it's an unusual circumstance, and it's unintentional that causes friction.
Yeah, I see.
But you're right, it's a potential gap.
I got a quick question for Lorrie, if there's anyone behind me. So back in the day, people would, FDA folks could send out slides. Someone told me that they were all supposed to go through OCOD now to ask for slides. Was I told correctly, or?
Essentially, it becomes a FOIA request because they're not provided. They get them on site at the meeting, that's totally fine. But after the fact, it becomes a FOIA request. Now, they would go into our fast track queue, so we turn them around very quickly. So, it would probably be a few days before we get to it.
Okay, a few days, that's great.
Right, unlike some of other FOIA requests that are complex that right now we might not get to for a couple of years.
Right, yeah. I tried to avoid submitting FOIA requests because I remember what they were like. But I will say one thing before I yield to the person behind me. The manufacturing assistance in OCOD is really helpful. And if it's not a CBER issue, they will reach out and find which center it is. If it is a CBER issue, they will find the right person, having been the recipient of a lot of questions from OCOD when I was there. So they work hard to get you the right answer.
Yeah, thank you.
Okay. I think we're going to have to draw things to a close, but before we do, Lorrie, I have one question for you because it's something I get all the time. When people come in with a speaker request, who should they send the email to?
We actually have a dedicated email account, which I should have included in my slides, and I believe it's CBER Speaker Requests, CBER Speaker. CBER Speaker Liaison at fda.hhs.gov. And you're looking at the CBER Speaker Liaison.
For the people online, she's pointing out Lorrie Warren Henderson, who was involved in the organization of the CBER track, along with Sherry Stacey Rivette, and they both did an excellent job. So I'd like to close this session now. I hope you would join with me in thanking Scott and Lorrie for great presentations. And we will be reconvening at 2:40 p.m. For the third session of the CBER track. Thank you very much.
So good afternoon and welcome to session 3. My name is Cecilia Crowley, as you can see on the screen. I'm the moderator for session 3, the last but not the least, covering part 2 of the regulatory communication. Our first speaker is Cara Pardon. She's is a regulatory project manager in the Office of Review Management and Regulatory Review in OTP at the Center of at CBER. Cara is a regulatory expert and project manager with a passion for protecting public health and safety. Since joining FDA as an RPM in November of 2020, Cara has provided review management throughout the product life cycle, from pre-submission meetings to post-marketing submissions. As an RPM, Cara also acts as a liaison between FDA and industry, managing all communications and meetings. Prior to her work at FDA, Cara served as a biologist at the National Cancer Institute, National Institutes of Health, and Cara received her Bachelor of Science from the University of Wisconsin-La Crosse and her Master of Science from Cornell University. Please welcome Cara Pardon.
Thanks, Cece, for the introduction. Good afternoon, everyone. It's very nice to see all of you here in person, and a hello to all of you attending virtually. My name is Cara Pardon. I'm a regulatory project manager, or RPM, in the Office of Therapeutic Products, OTP, within CBER. Today, I'll be providing a high-level presentation on types of regulatory submissions specifically reviewed within CBER OTP. By the end of this presentation, you should be able to identify and define the types of regulatory submissions in the biologics lifecycle under the PDUFA program, from interact meetings through BLAs and post-market submissions. You should also be able to describe the timelines associated with these PDUFA submissions. You should also be able to identify and define the types of regulatory submissions in the medical device lifecycle under the Medical Device User Fee Act, or MDUFA program, from Q submission meetings through pre-market applications and post-market submissions. And this presentation will also identify best practices and tips for submissions and interactions with OTP during submission review. Of note, this presentation is going to touch the surface on a lot of different topics, so we do have various resources available in the form of our guidances, our webpage, and our OTP Learn website listed at the end of this presentation for anyone who's interested in learning more. So first, what is PDUFA? PDUFA, or the Prescription Drug User Fee Act, was created by Congress in 1992 and authorizes FDA to collect user fees from persons that submit certain human drug applications for review or named in approved applications as the sponsor of certain prescription drug products. And since the passage of PDUFA, user fees have played an important role in expediting the drug review and approval process. And PDUFA must also be reauthorized every 5 years. The PDUFA submissions that I will be covering today include pre-submission meetings, IND applications, master file submissions, BLAs, as well as post-marketing submissions. Provided here is an overview of the PDUFA product lifecycle from preclinical development to post-marketing, which I will use as a reference going through. So first, I will review initial targeted engagement for regulatory advice on CBER products, or interact meetings. An interact meeting is a formal meeting with statutory timelines under PDUFA 7. Interact meetings are for novel therapies and products that may present unique development challenges. Interact meetings are for sponsors to obtain early feedback on the development of a specific pre-investigational product. So sponsors should not expect an interact meeting to help choose an investigational product or between products. And please note that only one interact meeting will be granted for the chosen product and specific clinical indication. Interact discussions will focus on choice of preclinical models, toxicology studies, and design of proof of concept studies. Chemistry, manufacturing, and control strategies to demonstrate product safety to support the product's use in first-in-human studies, as well as clinical trial recommendations for first-in-human studies in a specific clinical population. For an interact request, the meeting package must accompany the meeting request and should identify key issues and provide specific questions for discussion. CBER SOPP 8101.1 is a great reference to understand CBER meeting types, meeting request expectations, timelines, and the scheduling process. After submission and receipt of an interact request, sponsors should expect a meeting grant or denial decision within 21 days. This will be sent via email from the submission assigned RPM. And if the request is granted, the meeting may be scheduled as either a written response only, so this means no meeting will be held with the sponsor, a teleconference, this is a phone discussion only with the sponsor, a virtual face-to-face meeting, this is a Zoom meeting which will include cameras, or an in-person face-to-face meeting or hybrid meeting where OTP and sponsor attendees will meet at the FDA White Oak campus, and some attendees may also join virtually. If the interact meeting request is granted, the meeting will be scheduled within 75 calendar days or as close to 75 days as possible. If written responses only, the WRO would be provided by day 75 as well. For scheduled telecons or meetings, preliminary responses will be provided to the sponsor 5 days ahead of the meeting date. And the sponsor is expected to provide a response and final agenda at least 2 days before the scheduled meeting. An interact meeting will not have formal meeting minutes issued after, but if advice has changed, the RPM will provide annotated preliminary responses within 30 days of the meeting date. And a sponsor may also request clarification on WRO or annotated preliminary responses within 20 days, and OTP would provide response within 20 days of that request. Another meeting option for early interaction with OTP is a pre-investigational new drug meeting or a pre-IND. A pre-IND meeting is a formal type B meeting. And again, it's for sponsors to obtain early feedback. A pre-IND meeting should be requested when the sponsor already has a defined manufacturing process developed and has completed proof of concept studies and some preliminary and safety toxicology studies. A pre-IND meeting should be requested to discuss IND enabling CMC and PharmTox data, as well as clinical trial design. And as with interacts, specific questions for discussion should be posed. For a pre-IND meeting request, the meeting package does not need to accompany the request, but should provide a summary, background issues, and draft questions for discussion. And again, CBER SOPP 8101.1 provides further information on our regulatory meetings. After submission and receipt of a pre-IND meeting request, the sponsor should expect a meeting confirmation or denial within 21 days. Again, this will be sent via email from the submission assigned RPM. And if the request is granted, the meeting will be scheduled as one of the 4 previously mentioned meeting formats: written responses only, telecon, virtual face-to-face, or in-person face-to-face. If a pre-IND meeting request is granted, the meeting will be scheduled within 60 calendar days, or as close to 60 days as possible, and WRO provided by day 60. The final meeting package is due from the sponsor at least 30 days before the scheduled meeting. Preliminary responses will be provided to the sponsor 2 days ahead of the meeting, with a final response and agenda expected from the sponsor at least 1 day prior to the meeting. Formal meeting minutes will be provided for a pre-IND meeting within 30 days. And again, a sponsor may also request clarification on written responses or meeting summaries within 20 days after issuance, and OTP would provide response within 20 days of that request. Some best practices and helpful tips for interacts and pre-INDs. Firstly, please ensure that the point of contact has secure email established. A request for secure email can be sent to the email address listed on this slide. RPMs cannot communicate any regulatory information without secure email established, so we do recommend that sponsors have this set up before submitting a meeting request. Sometimes sponsors are unclear on if they should submit an interact or a pre-IND, so please consider the product stage of development. For example, to discuss manufacturing strategies and proof of concept studies aren't yet completed, an interact should be requested. If some preliminary studies have been completed, you may consider a pre-IND meeting. And of note, a sponsor does not need to hold both an interact and a pre-IND meeting for a given product. It may make sense to hold one or the other, or both, or neither, depending on the product's development stage. Please also ensure to respond to communications in a timely fashion to facilitate our review. And lastly, please consider our guidances and SOPPs, which detail the expected meeting package information and meeting procedures. For example, questions for discussion should be targeted and specific, not open-ended. And please also consider meeting agenda guidance, which would be provided along with our preliminary responses. The next submission type expected in the PDUFA development lifecycle is an investigational new drug application, or an IND. Under 21 CFR 312, any use in the US of a drug or a biological product not previously authorized for marketing in the US requires submission of an IND application to the FDA. An IND is submitted for an investigational drug to be used in clinical studies to collect safety and efficacy data. And any human research study must be conducted under an IND if the research involves a drug, is a clinical investigation, and is not exempt from the IND requirements. And with an IND, safety really is our priority. Our primary objectives in reviewing an IND in all phases of the investigation are to assure the safety and rights of subjects. And in phase 2 and 3, to help assure that the quality of the scientific evaluation of the product is adequate to permit evaluation of the product's effectiveness and safety. An original IND submission should include the following as specified under 21 CFR 312.23. Form FDA 1571, a table of contents, introductory statement, and general investigational plan, an investigator's brochure, the clinical protocols, CMC and PharmTox data, any previous human experience, and other relevant information for the product. The original IND timeline. Sponsors will receive an acknowledgement letter for the IND within 14 days of receipt. This will detail the submission tracking number, or STN, the assigned RPM, and some of the sponsor's responsibilities. There's a 30-day interactive review process. And by day 30, notification will be provided that either the study is safe to proceed, placed on clinical hold, and the study may not proceed, or placed on partial clinical hold, meaning certain aspects of the study may be allowed to proceed while others may not. Some best practices and hints for IND submissions. Again, please ensure that the point of contact has secure email established and is available for communications throughout the 30-day review process, BLA review. Firstly, a BLA involves an interactive review process, and requests for information should be expected throughout the review cycle. The applicant should also have any manufacturing facilities prepared for a potential pre-license inspection, as one may be required. And if in response to an information request, a substantial amount of new data, a substantial amount of new manufacturing or facility information, or a new analysis of studies not previously submitted to the pending application is provided, this may constitute a major amendment. Only one major amendment is allowed per review cycle, and a major amendment adds an additional 3 months to the review schedule. Applicants should also expect any post-marketing commitments or requirements to be communicated and discussed during BLA review, as well as labeling negotiations to agree on the final content of the package insert. And by the PDUFA action due date for the BLA, the applicant will be sent either an approval letter providing authorization to market the product, or a complete response letter. The application will not be approved and specific deficiencies will be identified. And a CR letter could also be sent earlier in the review cycle. If a CR letter is issued, the applicant will need to address all identified deficiencies and resubmit the application in its entirety within 1 year, though an extension may be requested. And an applicant may also request a Type A meeting to discuss the deficiencies in the CR letter before resubmission. Some general best practices and tips for a BLA. Firstly, please ensure the application is complete and not missing any required information. Again, ensure that the applicant is prepared for any required pre-license inspections. Similar to other submissions, please ensure timely communication with the assigned RPM. So please confirm receipt when requested and provide any updates on delays for informational responses. Please also ensure that Form FDA 356H is filled out correctly for your original submission, as well as any requests for information. So any submission during the original BLA review should be marked as an amendment to the original submission with reference to the STN. The submission tracking number system for BLAs, the first 5 numbers are your BLA number. The second numbers are for supplements to an application, so those are not used during original review. And the last numbers are for numbering amendments. So for example, this represents Amendment 6 to application 1, 2, 3, 4, 5. And please also ensure that all sections of Form FDA 356H are filled out, such as the cross-references, manufacturing facilities, orphan designation number, NCT numbers, and authorized representative information. And please ensure the submission type is accurately selected on the form, since it could have downstream processing impacts. Okay, so the BLA was approved. What kind of post-approval submissions are required after marketing approval? There are numerous types of post-approval submissions. I don't have time to detail all of them, but they're required for a number of reasons, including changes to an approved product. These submissions include manufacturing, labeling, and efficacy supplements, annual reports, product correspondences, any submissions related to PMRs or PMCs, submissions for risk evaluation and mitigation strategies, Pediatric Research Equity Act submissions, as well as promotional materials. Provided here for reference is a summary of types of manufacturing, labeling, and efficacy supplements and their timelines. All CBER OTP PDUFA submissions need to be sent to CBER Document Control Center, or DCC. And for information on how to submit to CBER DCC, you can reference the document provided here. Another helpful guidance for industry to reference also listed here. This details the submissions that are required to be submitted to CBER in an electronic common technical document, or ECTD format, via the FDA electronic submission gateway. And non-compliance to this guidance could result in rejected submissions. We do also encourage sponsors and applicants who are not required to submit electronically to follow the CTD in preparing submissions for our ease of review. Okay, so now that I've covered the PDUFA submissions most often received in CBER OTP, I'm going to move on to MDUFA submissions, or submissions for medical devices. As Dr. Lapteva noted this morning, CBER does also review certain medical devices. MDUFA, or the Medical Device User Fee and Modernization Act, was signed into law in 2002. Medical device companies pay fees to the FDA when they register their establishment and list their devices with the agency, and whenever they submit an application or notification to market a new medical device in the US. And these fees help FDA increase the efficiency of regulatory processes with a goal of reducing the time it takes to bring safe and effective medical devices to the US market. And like PDUFA, MDUFA must also be reauthorized every 5 years. The MDUFA submissions that I will touch on today include Q submissions, which are device meetings, investigational submissions, marketing applications to include 510Ks, de novo requests, HDEs, PMAs, device BLAs, as well as a mention of post-marketing submissions. Before I delve into the MDUFA development pathway, it's important that I first explain medical device classification. Medical devices are categorized into one of three classes, 1, 2, or 3, based on the risks and the regulatory controls necessary to provide reasonable assurance of safety and effectiveness. Class 1 devices are generally deemed low risk and are not intended for use in supporting or sustaining life or of substantial importance in preventing impairment to human health. Examples would include bandages and hospital beds. Class 2 devices represent a higher risk than Class 1 devices, and general controls are insufficient to provide reasonable assurance of the safety and effectiveness of the device. Examples would include syringes and mononuclear cell collection devices for therapeutic use. And Class 3 devices represent the highest risk, usually sustain or support life, are implanted, or present a potential unreasonable risk of illness or injury. An example would include tissue and cell processing devices used at point of care, such as a device for skin biopsy dissociation and wound repair, reapplication for wound repair. A CBER OTP reviews an array of medical devices for human cells, tissues, and other biologics. I'm not going to read this slide in the interest of time and everyone's sanity, but for your future reference to review the types of devices that we specifically review within CBER OTP. Provided here is an overview of the MDUFA device development pathway from preclinical development and discovery to post-marketing. So first, I will discuss Q submissions or Q subs. A Q sub is a request for feedback or a meeting for medical devices. A Q sub can occur throughout medical device development. It could be a pre-submission to discuss a medical device at an early stage of development. It may be a submission issue request to discuss an issue raised during review of another device application. Q subs may be informational meetings, requests for study risk determination to determine if the device is significant risk or non-significant risk, a request for breakthrough device designation or a meeting associated with a device that has breakthrough designation, or a part of the Safer Technologies Program or STEP. And provided here for reference are timelines associated with some of the Q subtypes. Next, I will introduce investigational device exemption applications, or IDEs. An IDE is required for an investigational or significant risk device to be used in clinical studies to collect safety and efficacy data for the device. There are several types of studies which may occur under an IDE. An early feasibility study, a traditional feasibility study, the pivotal study for the device, any continued access study or observational registries, other supporting or confirmatory studies, or other significant risk device studies. The IDE timeline. Sponsors will receive an acknowledgement letter for an IDE within 5 days of receipt. There's a 30-day interactive review process. And by day 30, sponsors will receive notification that either the IDE is approved, the IDE is conditionally approved, meaning there are some concerns which need to be addressed and part of the study may be on hold, or the IDE is disapproved and studies may not begin with the device. And please note the difference between an IND, which is allowed to proceed, versus an IDE, which must be approved. So an IDE is an investigational device application. Now I'm going to move on to touching on some pre-market submissions, starting with 510K submissions. A 510K is a pre-market notification for moderate risk devices to allow a device to enter interstate commerce by demonstrating substantial equivalence to a predicate device. So what is substantial equivalence? A subject device is substantially equivalent to another legally marketed predicate device if it has the same intended use and same technological characteristics, or the same intended use and different technological characteristics, and there are no different questions of safety and effectiveness. And a 510K provides information to demonstrate that the subject device is at least as safe and as effective as the already legally marketed predicate device and substantially equivalent. A 510K decision is not an approval. A 510K may be cleared, meaning substantial equivalence has been demonstrated with or without limitations and may be marketed, or a determination that the device is not substantially equivalent and may not be marketed. De novo requests are another type of pre-market device submission. A de novo is a risk-based classification process. It's used for new devices whose type was not previously classified or no legally marketed predicate device exists. So any new device is automatically considered class 3 unless it's reclassified into class 1 or 2 through the de novo process. And a de novo may be declined, meaning the device remains in class 3 and cannot be legally marketed. And in this situation, the de novo requester should generally either submit a pre-market approval application or collect additional data and submit a new de novo request. If the de novo request is granted, a new classification regulation for the device type is established. The new device is authorized to be marketed and must be in compliance with applicable regulatory controls. And the new device may now serve as a predicate device for 510K submissions of future devices of the same type when it's applicable. Next, I'll define a humanitarian device exemption application, or an HDE. An HDE is a marketing application for an HUD. An HUD being a medical device intended to benefit patients in treatment of a disease or condition affecting manifested in not more than 8,000 individuals in the US per year. And the goal of an HDE is to demonstrate reasonable assurance of safety and probable benefit. An approved HDE is exempt from the effectiveness requirements of sections 514 and 515 of the FD&C Act and subject to certain profit and use restrictions. Decision options for an HDE application are approval order. The HDE is approved and the device may begin commercial distribution. Approvable letter. The application is placed on hold for minor deficiencies or the need for a GMP inspection. A not approvable letter identifying major deficiencies that still need to be addressed. And lastly, a final action of denial order. The HDE is not approved. Next, a pre-market approval application, or a PMA. A PMA is required to evaluate safety and effectiveness of the highest risk medical devices, class 3. It's the most stringent type of device marketing application, and general and special controls alone are insufficient to assure safety and effectiveness, with the ultimate goal being to demonstrate safety and efficacy of the device. The decision options for a PMA are the same as for an HDE. Approval order, the device may be marketed. Approvable letter, there are minor deficiencies that need to be addressed. A not approvable letter, there are major deficiencies to address. And a denial order, the application is denied and the device cannot be marketed. Before I move on to types of device post-market submissions, I'd also like to mention that CBER OTP may also review a device BLA, which follows the PDUFA pathway, but may include Q-subs, a device IND, and followed by a BLA for the device. So just like for PDUFA products, there are also device post-marketing submissions. A 522 order may be required for post-market surveillance studies for 510Ks, HDEs, PMAs, or de novos. 510Ks may also have post-marketed changes being affected or recalls, corrections, and removals. PMAs, HDEs, and device BLAs may also require supplements and annual reports. And device BLAs may also be subject to PMR and PMC submissions. Some general MDUFA submission guidance for CBER OTP. Again, please ensure that secure email is established. Medical device submissions to OTP are required to be submitted to CBER DCC in an electronic copy format or e-copy format in accordance with the e-copy guidance. Many medical devices are also going to be subject to these MDUFA user fees. For a CBER OTP submission, please ensure that the user fees are directed to CBER and not to CDRH. And lastly, as of October 1st, 2023, 510K submissions require the use of an eStar format. This is an interactive PDF form to guide applicants through the process of preparing a medical device submission. Okay, are you all still with me? We have covered a lot today, both PDUFA and MDUFA submissions reviewed within CBER OTP, as well as a number of tips and best practices. So we will move on to a few challenge questions. Challenge question 1, which of the following statements is not true? A, CBER master files are reviewed and approved as independent submissions. B, to cross-reference a master file in your IND submission, a letter of authorization must be included. C, a BLA filing notification decision will be provided within 60 days of receipt. Or D, secure email is required for regulatory communications. So I'll give you just a few seconds to think about it. So which of the following statements is not true? The answer is A, CBER master files are not reviewed nor approved as independently from investigational or marketing submissions. Challenge question number 2, original IND and IDE submissions have a decision timeline of A, 60 days, B, 90 days, C, 30 days, or D, there is no specified timeline. And I hope this will be a quick one, so I'll give you just a few seconds. Original IND and IDE submissions have a decision timeline of 30 days. And our last challenge question, number 3, which of the following statements is true? A, a PMA is required to evaluate safety and effectiveness of low-risk medical devices. B, an HUD is a medical device intended to benefit patients in a disease or condition that affects more than 8,000 individuals in the US per year. C, a 510K is required when introducing a moderate risk device to the market for the first time if there is a predicate device. Or D, a de novo submission may be approved or conditionally approved. So again, I'll just give you a few seconds to think about it. So which of the following statements is true? The correct answer is C. So for A, a PMA is required for high-risk medical devices, not low-risk. B, an HUD is for a disease affecting less than 8,000 people in the US per year. And for D, a de novo request is either granted or declined, not approved. So in summary, OTP reviews a wide array of regulatory submissions under the PDUFA and MDUFA programs. There are many chances for interaction with OTP throughout the PDUFA and MDUFA product life cycles, and there are common best practices for regulatory submissions and communications with OTP. So we've covered a lot today, so there are many resources on the next couple of slides for your review. Lastly, CBER OTP looks forward to working with you during all stages of your product development. My contact information is provided here, as well as our website and our OTP Learn webinar series, if you'd like to learn more. With that, thank you very much. I will be taking questions after the next presentation.
Thank you, Cara, for that presentation. So our next speaker is Jackler. Karen is a program manager for the CBER Patient Engagement Program. She works with patient organization with her FDA colleagues to coordinate patient engagement activities and to facilitate information sharing and collaboration. Ms. Jackler joined FDA in 2009, and prior to joining CBER in 2018, she served as a strategic communication advisor and project manager for the FDA Center of Drug Evaluation and Research and FDA Center for Devices and Radiological Health. Her experience also includes communication research and outreach roles with the NIH and CDC. She holds a Master's of Public Health with a concentration in health communication and education and a bachelor's in cellular and molecular biology. Both degrees are from Tulane University. Please welcome Karen Jackler.
Thank you. Can you hear me? Hear me? I'm a lot later on. So yes, my name is Karen Jackler, and I'm the Patient Engagement Program Manager at the Center for Biologics and Evaluation. And I've been in this position since 2018. And I just want to thank the SBI organizers for having me here today to talk about patient engagement at CBER. My typical audience is patient advocacy groups, and it's especially nice to be able to talk to you, industry, today about patient engagement. All right, so my learning objectives are to first define patient engagement for you all, to describe how CBER considers patient input in the project review. And so to do that, I'm going to show you where to find patient information on the FDA website. And finally, I hope that in the short time we have together, that you will feel that you can begin to integrate patient engagement into your drug development program. I know there's not that many people here in the room at least, but before I move to the next slide, can I get a quick just show of hands for people in the room? How many of you have been engaging with patient advocates? Oh, perfect, great. So a couple of you. So hopefully you'll have the best questions for me later, right? Great. So what is patient engagement? So FDA has a definition for patient engagement, and it was developed as part of the patient-focused drug development guidances that I'm going to tell you a little bit more about later. So patient engagement activities are activities that involve patient stakeholders sharing their experiences, perspectives, needs, and priorities that help inform FDA's public health mission. So these activities can include things like testimony at advisory committee meetings, submissions to regulations.gov public dockets, meetings attended by patients, the FDA, and other stakeholders, other correspondence with the FDA, interactions through social media, and interactions with or information from patient representatives or patient advocates. So these are not the only activities for engaging with patients, but the key here is that the information is coming from patients in some way. There's no filtering. They are part of the conversation that is going on around drug development. And patient engagement is the first step in a patient-focused drug development program. To differentiate between patient engagement and patient-focused drug development, I'm not going to dwell too much on this. It actually could be an entire course. But patient engagement are these activities. Patient-focused drug development is taking the sort of initial engagement and developing in a systematically integrating this information to ensure that patients' experiences, perspectives, needs, and priorities are captured and meaningfully incorporated into drug development and evaluation. So there's the link for the patient-focused drug development glossary, which would talk about both engagement, patient-focused drug development, and all the other parts of patient-focused drug development. So what can you learn by engaging with patients? You can learn the impact of the disease and its treatment on the patient. This can include signs and symptoms of the disease or condition, the chief complaints of the patients. What is most bothersome to them? The burden of living or managing a disease or condition. This can include the effect of the disease on their daily activities of living and functioning. What is the burden of the treatment? And how does their treatment affect their daily life, their daily living and functioning? Sorry, I got to catch up with myself. We can also learn about patients' perspectives about potential and current treatments. What are their expectations of the benefits? What is their tolerance for harms or risks? What are acceptable trade-offs of benefits and the risks? That sort of falls under the umbrella of patient preference. What are the attitudes towards uncertainty about a treatment? We can also learn about their views on unmet medical needs and available treatment options. So where are current treatments falling short? How are they falling short? Are there segments of the patient population that are not benefiting from a current treatment? Oh, I went backwards. Go forwards. And by engaging with patients, we can also enhance the understanding of the natural history of the disease or condition, including its progression, severity, and chronicity. So when to engage with patients? So in short, throughout the medical development process, it's not a one-time thing. It's really part of the whole process of developing a product. So patient, you can collect it at the beginning, at the launch of a discovery program, or it may be independent of any specific medical product program. Data can be collected in a variety of settings, including clinical trials, observational studies, advisory boards, public meetings such as the FDD, patient-focused drug development meetings, which I'll talk a little bit about more later. On other novel settings, as I mentioned, social media and online patient communities. So Cara did a great job of socializing you to this timeline already, and I have my own things to say about this. So here's a representation of a typical medical product development timeline. And I'm going to take you through how engagement with patients could inform drug development at each of these stages. So the preclinical stage. Engagement can tell you what impacts, such as the burden of disease and the burden of the treatment, matter most to patients, and how do we measure them? So this information can inform endpoints and how to measure them. It can help you figure out early on which direction to take a program. I'll note that many patient advocate groups have taken on the task of developing information in this preclinical space. They've taken on developing registries or starting natural history studies. They've initiated research programs to develop animal models. They've worked with clinicians to develop standards of care, and they're developing clinical outcome assessments. So in my experience, patient groups are not sitting around waiting for treatments, waiting for product developers to develop these treatments. Many of them out there are laying the groundwork for biologics developers. So looking in the next sort of chunk of the timeline, clinical trials. So we're nothing without clinical trials, and we're nothing without the people who participate in them. So participating in a clinical trial can be life-changing for a person with the disease or a condition, and their participation can change the lives of people like them down the line. But participating in a clinical trial can be very burdensome on the patient. Many CBER products are for rare diseases, and thinking about the patients I've met over the past 5 or 6 years, patients have mobility problems. They need extensive durable medical equipment, and their medical schedule is very regimented. So I'll just sort of give you an example. So yesterday I was listening to a patient-focused drug development meeting with people and caregivers of people with proximal disorders. So proximal disorders are inborn errors of metabolism, and they cover a continuum of degrees and severity of disease states and different diseases. And people with paroxysmal disorders have seizures, vision loss, hearing loss, low muscle tone, developmental delays, feeding issues, and a host of issues affecting the brain, liver, kidneys, and bones. Most children are very late walkers. Most are eventually wheelchair bound. Many die in infancy. Parents and people with this disorder have to build and manage a lot of supports around their kids and themselves. And they have to see specialist physicians constantly, go to physical therapy appointments, speech therapists, audiologists, and many don't live near major medical centers, much less a center that might happen to host a clinical trial for their disease. So some of the things I heard them say about clinical trials is that to really consider what is absolutely necessary. What is it that matters? What is it that we really need to measure? What is necessary, but what can be done nearer to the patient? One of the examples they gave was a blood draw, having to take this child with all this equipment and upsetting the entire, all this information I just told you about their feeding issues and the mobility issues and all the appointments that they need to attend just to keep up in their little world. To go to drive an hour or two for a blood draw, do the blood draw, and then come back and have to drive another, that was really difficult for them to do. So could something like that be done closer? So the whole idea with getting patient input, engaging patients on clinical trials, is that to consider ways that you can tailor procedures to the patient population and to consider all the things that they have to go through to get to a clinical trial. And also, not only that, but also thinking about their experience when they're in the clinic. I'll just want to tell you another anecdote I heard, and it's really, so it was for another kid's condition, gave them very poor impulse control, which, a lot of young kids do that, but it's particularly exacerbated in different diseases. And so the child revolts if the mom let go of the hand to do something as simple as sign a document. So her point was, why not just think ahead about those things and make it less burdensome just to sign a document by setting aside a quiet room. So they're with the door closed. So you're not, signing a document and then chasing the kid down the hall. So engaging about clinical trials is not just about providing transportation, hotel rooms, or meal vouchers. It's about making, but it's also not about making a clinical trial Disney World experience. It's really about working with the patient and engaging them to make it workable so they can hang in there and you can get good quality data. At the marketing application, so submitting patient-reported data or patient preference data with the BLA is something FDA will consider as part of the overall application. And truth be told, developing the patient-reported outcome measurements and the patient preference studies is something that should start earlier, not when you're submitting the marketing application. And what I'll sort of note here about PROs is there are a number of standardized patient-reported outcomes out there, but from what I hear from patients about standard PROs, for say something like cognition, they may not measure what matters to that patient population. If the patient population is starting below the minimal measure that a PRO is intended to measure, it may not pick up the changes that are meaningful to that patient. And that's why early engagement is important. So I just talked a lot about how patient engagement can help tailor clinical trial protocols, but engagement is also important to making sure that a clinical outcome assessment endpoint corresponds to something that is a meaningful change to the patient. So after the drug is on the market, post-marketing. As you know, your job and the FDA's job does not stop with products on the market. So we have to continue to assess the safety of the product. In some cases, such as gene therapies, patients will need to be followed for a number of years, and just like with clinical trials, engaging with patients to inform what a follow-up study can look like. And when new information about a treatment becomes widely available in the post-market, that new information needs to be communicated to patients and prescribers and the people who have that treatment or who are caring for people with that treatment. And so engaging with them can help inform what those communications should look like. Who should they come from? How should I get the information? What are the information channels that matter to them? So now I'm going to move on to how FDA does patient input. So if any of you guys are, I'm sure a lot of you are familiar with the BLA principle, part of the publicly available documents that FDA makes available after an approval. And so there's a section here in the table of contents that I've highlighted called patient experience. And this memo is both from the CBER, Center for Biologics, and the CBER, Center for Drug Clinical Review memos. So this section was in response to the 21st Century Cures Act of 2016. And I'm just going to go a little wonky here. And so under the 21st Century Cures Act, FDA is required to make public a brief statement regarding the patient experience data and related information, if any, submitted and reviewed as part of the application. And so section 1.2 addresses this. So let's take a closer look at section 1.2. So the biggest feature of section 1.2 is this table. So the top section includes the range of patient experience data that fits in the 21st Century Cures Act definition of patient experience data that might be submitted by the sponsor. And the bottom could include other patient experience data that a CBER review team might have considered because they were aware of it. And so if you kind of, I'm not going to go one by each of those, but you'll see that there are different sort of quantitative outcome measures such as COAs that measure how patient feels, function, or survives. There's descriptive data such as PFD meetings and listening sessions. Qualitative studies like focus groups and interviews, surveys, natural history studies, patient preference studies. So what you'll see, and I'll show you a couple of examples, is that the review team will indicate on the left side what patient experience data was submitted or considered. And on the right side of the table will say where in the memo that information is discussed. The review team can also add in this section some narrative about their thoughts on patient experience data. So let me take you through a couple of case studies. That show you how CBER has considered patient experience data in the context of the clinical review memo, as well as a couple of other ways that patient experience, that patient engagement has been integrated into some of our activities. And before I start into these examples, please don't take my descriptions of these uses or anything as regulatory truth. I'm not a reviewer, and my goal here is to get across the importance of patient engagement, and I'm taking some liberties here to make this more conversational. So please use the FDA website to get more accurate information about these products. Okay, so the first example I'm going to talk about is purified allogeneic islets of Langerhans for transplants. The trade name is Lapteva. So this is a cell therapy to treat type 1 diabetes mellitus in patients who are unable to achieve target glycemic control because of the ongoing risk of hypoglycemic events. So this is for patients who have difficulty to control diabetes. So despite using insulin, monitoring, education, they haven't been able to control their diabetes. And they also have developed hypoglycemic unawareness, so they can't detect when they're experiencing insulin so low they might be consciousness. So for some patients, Donacell, which is the FDA keyword, can help patients produce insulin on their own and may make them insulin independent. But the transplant procedure presents significant risks, and it may not work for every patient, and it may not last for all of the person's life. allogeneic transplants need immunosuppression, which has its own risks. And so these circles here highlight how FDA considered patient input. So here we have the narratives from section 1.2 that I was talking to earlier. So the applicant submitted testimonial from the Donna Stultz advisory committee meeting, and the reviewers considered the results of the patient preference study on islet cell therapy. The patient preference study looked at what level of risk of serious complications patients are willing to accept for a certain extent of insulin dependence, and that helped shape the benefit risk assessment for people. My next example is Limogene Gepropavic SVDT. The trade name is Biologics. This was approved for the treatment of wounds in patients 6 months and older with dystrophic epidermolysis bullosa. So epidermolysis bullosa is a genetic disease that makes them very fragile. There's an absence of a collagen that binds the layer of the skins together, of your skin together. And blisters will appear in response to the slightest injury, such as rubbing or scratching. These symptoms begin at birth and can lead to large painful sores on the body, and managing the disease is very difficult, and there can be many complications, such as malnutrition, anemia, infection, and skin cancer. So this drug is a topical vector-based gene therapy that can express the collagen that the patient was missing. So here you can see that the sponsor submitted patient-reported outcomes and observer-reported outcomes as part of the application. So the review team checked that it was submitted, and they put on the far right the section where that information was discussed. What was also interesting about this is that the FDA staff also considered direct patient input in the form of a listening session that the review staff for this product requested. So an FDA listening session, I'm going to talk a little bit more about it later, but it's a program where FDA review staff can hear directly from patients when the review staff has questions for patients that they think can inform their regulatory questions. In this case, it was understood that the product would not completely close all epidermolysis bullosa wounds, but they wanted to understand what level of wound healing, wound area reduction would represent meaningful improvement in pain and in management. And the FDA heard from patients that even partial wound closure was important as it reduced pain and burden associated with wound care. And that became part of what was considered, along with all the other clinical data that was submitted by the So I have a couple of other examples. These are not from the clinical review memos, but from other public documents. This one is for a gene therapy for metachromatic leukodystrophy called MLD. Called I should have practiced before. Adadars. Well, I'm sorry. I am not going to be able to pronounce it, but the trade name is Lameldi. But anyway, so MLD is a rare autosomal recessive lysosomal storage disorder, primarily due to a deficiency of aryl synthetase A enzyme, or ARSA enzyme. ARSA deficiency leads to widespread demyelinization of the central and peripheral nervous systems with severe progressive neurologic impairment and ultimately death. And so it affects the brain and the nervous system, and impairs mobility and cognition. As the disease progresses, caregivers describe their children are leveling with MLD as becoming locked in and unable to communicate. They can't smile. Any word they had, they might have lost along the way. They can't try gestures in response to questions. So that was something that was defined. Not only was mobility important to the families that are caring for these children, so was cognition, because that was how they could tell their kids were doing okay. So this is one of the supporting documents that's posted on the FDA webpage for this approval. And it's a document called BLA Clinical Outcome Assessment Review Memorandum. And section 3 is on patient experience data. So this MLD drug, MLD, was approved for 3 subtypes: pre-symptomatic late infantile, pre-symptomatic early juvenile, and early symptomatic early juvenile. So the primary endpoint was severe motor impairment-free survival. But it also showed that pre-symptomatic late infantile children treated with the therapy had normal language and IQ scores. And in addition, children with pre-symptomatic early juvenile and early symptomatic early juvenile MLD showed slowing of their motor and cognitive or their cognitive part of their disease. So you can see here how patient experience about preserving neurocognitive function, even if the patient was experiencing symptoms already, was very important to the MLD community, and how this was considered in approving the therapy for all subtypes and not just pre-symptomatic patients. One more quick example. So this is another example of how an FDA-led listening session was incorporated into FDA's thinking. So this was not necessarily considered in the review for featured hemophilia products, but the patient engagement did influence scientific conversations. So FDA hematologists led a listening session with people with hemophilia and their caregivers. And what we heard was there was consistent interest in understanding the benefits and risks associated with gene therapy and receiving accurate and complete information about gene therapy and hemophilia. They acknowledged that aspects of enrolling in clinical trials can be burdensome. The frequency of the visits to a clinical site were not a top concern. And the definition of successful gene therapy varied. So following the listening session, the input shaped the scientific discussion in a couple of ways. So it shaped the input for the product development and hemophilia product public workshop in December 2018. And at the meeting, there was further patient perspective heard from patients on a panel on patient-reported outcomes, particularly how instruments that have been used in clinical trials didn't really measure what mattered to them, or questions didn't make sense to them. And so that also became part of the scientific conversation around what a clinical trial for hemophilia look like. And so this is the sort of final case study I have on hemophilia illustrates that the FDA values patients being part of the scientific discussion, informing the developers, regulators about what matters to them and how that can be incorporated into the drug development program. All right. So, Cambridge. Okay. Challenge question 1. In which ways can patient engagement not help your biological development program? Developing clinical outcome assessments, tailoring clinical trial protocols for patient needs, persuading FDA to reward your history, or understanding patients' expected benefits and persuade us to miss your but they can help in developing COAs because patients can tell you what matters to them and what aspects of the disease bother them most, which is the start of systematically incorporating them into measurable outcomes. Clinical trials, yes. Patient engagement can help you tailor your clinical trials so that it's less burdensome to patients, which can contribute to enrollment and retention. And yes, patient engagement can tell you about their understanding, can help you understand expected benefits and tolerance of risks. It can, and that can, which can also be systematically incorporated into patient-focused drug development programs. Okay, so we actually have some resources, and I'm just going to take you through a couple of the websites where these resources are. But first, I'm going to take you through a couple of patient engagement programs. The first are patient-focused drug development meetings. So these are meetings that provide key stakeholders, including the FDA, patient advocates, researchers, drug developers, healthcare providers, and others, an opportunity to hear the patient's voice. And the lessons learned from these meetings include, but are not limited to, specific experiences that matter most to patients, patient perspectives on meaningful benefits, and a lot of the other things that I've already talked about. I see I'm running low on time, so I'm going to speed up a wee bit. And after these meetings are almost always followed up with a voice of the patient report. Patient listening sessions help the agency inform medical product development, trial design, patient preferences, and shape our regulatory thinking. During a patient listening session, patient staff will either ask questions or simply listen to better understand patient experiences and perspectives. And these are followed up with a brief summary of what's available, of what was said at the meeting. RegenMed Ed is a program that from the Center for Biologics Office of Therapeutics and Products. It stands for Regenerative Medicine Education. And so this series, it's a series of programs that brings together important stakeholders and the FDA to discuss foundational information about regenerative medicine therapy. And most importantly, it includes patients right there in the middle. It's talking to patients. Patients are talking as well. People are learning from one another about how patients and patient advocacy groups can move drug development forward. CBER's OTP also has workshops, meetings, and town halls that engage with product developers and researchers. And there are webinars on topics like CAR-T therapy, human genome editing, capturing post-approval safety and effectiveness data for gene therapy, and many of these include the patient perspective. So, that's right. There are reports, recordings, and transcripts. They're available to you. So I'm going to take you through on a quick tour of a couple of these websites. Okay, so the first one is the voice of the patient in listening session reports. There are 195 reports on how patients experience their disease, all from the patients. So the audience for those reports is all people with an interest in drug development, and FDA reports include all the materials associated with the meeting. I'm going to yeah, sorry, I clicked on this. So what I'm going to show you is that all the reports are available. They are alphabetized, and you can search for them here. So, do you mind helping me to go up? That's all you need. So up pop's the patient listening section here. And this leads to the summary there. Another one. So MLD, I showed you the metachromatic leukodystrophy. Here's the PFDD meeting that was referenced in the example that I showed you. And then sometimes patient groups will get together and talk about their diseases as a group. One example is DPP. So developmental epileptic encephalopathies. So sometimes patients work together and there may be a general topic in there for people. So just sort of think about how patients think about their disease when you're looking at things there. All right. Join that up. Okay. There are 22 events related to Genome. This includes 7 meetings that featured patients or how patients can engage in the development of regenerative medicine. So, go down here. Check out. So all of these lead to more information. It includes information about the meeting, and as these meetings have been completed, oh, this one's coming up. Look at that, guys. Register now. So here's a either clinical trial information. So there's a recording available, and the event materials are down. And there's a complete transcript, so you can search for information inside the transcript. And the last one. And then I just want to quickly take you very quickly through the patient-focused drug development guidances. So these guidances address, hopefully you're familiar with these, but if not, the patient-focused drug development meeting addresses in a stepwise manner how stakeholders can collect and submit patient experience data. So guidance 1 discusses sampling methods that can be used when planning to collect patient input, and it provides a general overview of the relationship between potential research questions and methods, and when deciding from whom to get patients to collect input. Guidance 2 takes it a step further. So what methods can you use to elicit information from the individuals that you identified in guidance 1? Guidance 3, closer to COAs. It helps ensures that clinical outcome assessments used during medical product development measure what matters to patients, are clear about what is measured, are appropriately evaluate the effectiveness, tolerability, and safety of treatments. And then guidance 4, how do you put those COAs into a clinical trial? So, so the website has all of these. They're organized right here, all together for you. So you can. It's like a little story here. It can tell you, like I said, it's stepwise. So they're all here, and so as you're kind of moving through your program or trying to identify where you are in your program and how you can use the patient experience, how you can turn that into something that's measurable, this website can take you through that. Take you to the most current part of the guidance. Some of the guidance are still in draft, but they're moving forward. Okay, so I'm just a few minutes left, and which means it's time for the next game break. What patient engagement resources are available on the FDA website? A list of almost 200 reports and resources, a matchmaking service for drug developers and patient groups to collaborate, stepwise guidance on collecting and submitting patient experience data, publicly available documentation on how FDA considers patient input. So A, C, and D. Yes, those are available. I took you through all of them. We do not have a matchmaking service, but I kind of put this in here because it gave me an opportunity just to emphasize again that there are a lot of patient groups up there who are doing a lot of good work and can be good drug development partners. We can't match you up with people, but there are a lot of organizations out there who can help you find patients who are willing to be good partners who are doing really good work. So these are some of the websites that I took you to today. And to keep up with patient engagement events that are led by CBER, we have a listserv, and or follow us on our social media accounts. In summary, I just want to leave you with the idea that patients are experts in what it's like to live with their disease. So talk to them. Patient engagement is the first step in patient-focused drug development. And FDA has resources to guide you. I showed you a lot today, and hopefully that gives you a little bit of information to help you see how it can fit into your program. My closing thought is engage with patients early and engage with patients early. So thank you.
Thank you, Karen. So now we're in the Q&A session, and the mic is open for anybody that have questions while we wait for Cara to make it to the stage. Anybody?
Hi, Hisan Smolder from Biologics Consulting Group in Alexandria. I have a question for Cara. We interact with FDA regularly, and our clients typically ask us to reach out to the RPM and ask questions and whatnot. And generally, we try not to bother you guys. But when there's an RPM that's not very responsive, what's the best course of action? Like, what do you recommend?
Yeah, so if an RPM is not very responsive, hopefully that doesn't happen often. We do try to be as responsive as possible. We do have a heavy workload, so sometimes it can be hard to respond right away. Hopefully it's not because the RPM doesn't work here anymore and you're not contacting the right one. But we do have an RPM main email that was listed at the end of one of my slides. You can always reach out to that line as well if you're not able to get a response. But again, hopefully that doesn't happen too often.
So there are a few RPMs that we have issued with, so we typically know, which one they're responsive or not. So I just wanted to know what the next.
Yeah, I would recommend our main RPM email.
I've got a couple of questions. Cara, type D meetings are pretty new. Do you have any comments on experiences with agents had in type D so far and suggestions maybe to make them useful from the sponsor's perspective? I know they're supposed to be short and focused, but other than that.
Yeah, so type D meetings started with Gopa 7. I think that was the beginning of last year. I think there was actually a REdI presentation last year. You can probably find the recording on type D meetings, but yes, they should be specific, targeted to one, a focused area, such as for CMC, like comparability or potency assays, common themes that we see with type D meetings. So we are having type D meetings since we started having them. But again, I'd recommend reading our guidances as well for further information.
So do you need to have an IND to have a type D?
I will pass on that question. I've not seen that. That's typically not what we would see. But
yeah. I also want to say, relatively recent, some of my clients have experiences in the 30-day review period, and the clients got a lot of IRs, which don't freak out. And I'm like, oh, wait, they're trying to get clarifications. What do you mean? Writing old comments is difficult to get an IR. and get past that. So I want to thank you guys for doing that. I know it's work, but it was really helpful for my clients to sort of see those IRs and see that the agency is trying to get some verification at the time to not put people on hold.
Yeah, exactly. The IND process is interactive in that 30 days. So we're sending those IRs to avoid putting anything on hold if possible, trying to work out any issues that we can work out in those 30 days. But yes, I agree. It can be scary to see some of those comments, but it's for the best of everyone trying to work those out.
See, I'm going to sit down yet. Karen, I love talking to patients and advocacy groups, and we do a lot of that. And maybe they're in the guidances you decided, but and I understand the situation, the good examples you had with the advisory committee meetings and the BLA summaries. Do you have any suggestions on how to work that into an interact or into an IND in a situation, particularly when you're trying to think about how to get patient-reported outcomes in clinical trials early on?
So it sounds like is your question if patients can be at an interact meeting, or how to like bring that into the early discussions?
How to bring that into the early discussions, because that's something that would be helpful. I'm not really sure how to approach that.
I don't have anything specific that I could advise there. I think, I think really understanding where the patient group that you're working with is, what they have already achieved. Sometimes they've already, patient groups are really good at knowing where each other is globally. That's something that social media has been really helpful with, is establishing those, at least a patient registry. Patients have taken on the role of developing their animal models so that developers can sort of do that, that proof of concept. Yes, we can, like, a gene therapy, we have done this on an animal model. I think where you might start with the patient groups in those early stages is really defining what success means to them, what is effectiveness. And maybe that's a good place to start those early conversations, is understanding, like I said, what's important to them, what do they think a successful clinical trial would look like? What do they think? What would keep them from participating in a clinical trial? So really sort of starting with really understanding where the patients are in those early days.
I appreciate that. Maybe talk about what patients are willing to accept at risk. That you might, if you weren't in that situation, you might have distinguished risk-benefit ratios. That would kind of bring that into the discussion.
Yeah, I think those early discussions, you can start talking about that. I think the very first thing you need to understand is what patients value, and then you can start figuring out once you understand what they value in a treatment, then you can start figuring out sort of what those like trade-offs are. Like I would trade 5 years of insulin independence for these allografts, those sorts of things. But maybe that's not it, but maybe it's really trying to understand what those are, what those trade-offs are. So
I appreciate that. I appreciate that I'm not the only one that can't pronounce proper names of drugs. Yeah, and every syllable means something, but they're not meant to be pronounced in English. Thanks.
Thank you. Any more question on the floor? Go ahead. You look like you have a question. Okay. Okay. Yes. Any online questions?
Yes, this question is for Cara. What are some common denial reasons for Interact and pre-IND meeting requests?
Yes, so some common Interact and pre-IND denial reasons. For an Interact, if the meeting package isn't included, that's common denial reason. The meeting package needs to be included. It may be too early in the stage in product development. Again, an Interact is not for us to help decide on what product you want to use in trials. It may be too advanced a product development. In that case, you may consider a pre-IND meeting. For a pre-IND, similar situations, it may be too early for a pre-IND. Haven't completed any preliminary safety toxicology studies. An Interact may be more appropriate. A pre-IND should also be multidisciplinary. I didn't really touch on this, but we typically tend to only grant one pre-IND for a specific product and indication, even if there's a change in sponsorship. So we want that to be a multidisciplinary meeting. And a pre-IND shouldn't be submitted to pre-review an IND. So there needs to be specific targeted questions. So those are some common reasons that we might deny either of those meetings.
Any additional questions?
Yes, Karen, this question is for you. How can regulators and sponsors improve how they listen to and respond to patients' current needs when it comes to perceived risk-benefit ratios for a specific drug in development, especially in cases where, due to heavy disease burden, patients may be willing to accept a higher risk?
Can you repeat the question?
Sure, Karen. So the question basically was stating, how can regulators and sponsors improve how they listen to and respond to patients' current needs when it comes to the perceived risk-benefit ratio of a drug in development, especially in cases where, due to a heavy disease burden, patients may be willing to accept a higher risk?
How to improve how we listen. That's a really, I guess I'm not quite sure to answer that question, but I think one of the things I would say is that there are lots of opportunities in sort of what we're seeing now is early collaboration and partnerships with patients and regulators and sponsors, where folks are talking about risks and benefits, and really, trying to work out where are the gaps in the information that can help us turn that into something that can be measured. And so I think that's a fantastic question, and it's not something that I think I can answer fully here. But I think part of the answer is really is that early collaboration and really engaging patients in terms of and all the different stakeholders to identify where the gaps are and to understand the burden of disease and the risks and benefits. And it sounds like, even inside a patient population, there could be different needs, different unmet needs, and different disease burden. And so really understanding all those different segments. So I'm not sure that I answered the question, but that's sort of where my current thinking is right now.
Thank you. Do we have an additional online question?
Yes, this question is for Cara. Does FDA consider the requested format of the FDA interaction in considering whether or not to grant a meeting? More specifically, if a sponsor who may have limited funds to support FDA meetings but wants FDA advice requests a written response, would that potentially drive FDA to deny a meeting?
That's a good question. So we consider the requested meeting format, but it's not a reason that we would necessarily deny a meeting. So if written responses only is requested, that wouldn't be a reason that we deny the meeting. We may, if it's a significant meeting, like a pre-BLA meeting, we may suggest a different format of meeting, like a face-to-face meeting. And it goes the other direction too. If you request a face-to-face meeting, virtual or in person, and we grant that as written responses, we could change the format, but we're not going to deny it just because of the format that you're requesting.
Okay, do we have more questions?
Yes, Karen, this is for you. Another awesome question. What does it take to see effective patient engagement? And what are some best practices and lessons learned to achieve this?
So what does it take? What does effective patient engagement look like, and what are some best practices is what I'm hearing. I think effective patient engagement is patient engagement that comes early, that can be integrated into the process. A lot of times, patient engagement is sort of maturing, I guess, in a way. I think a lot of times we see patients coming to us later, or sponsors bringing patients to us sort of late in the process. And having a PFDD or a listening session late in the process is not going to get the same results as having that early in the process, when you can really think about what you've heard from the patient and how those things that they've told you that matter to them can be part of the drug development program. So I think the key to effectiveness is trying to figure out is working with them at all stages, not sort of picking them up and putting them down, really making them part of the discussion and integrating that into the process. Different patient groups patient groups are on a lot of different levels, and some are, like I told you, some have started developing registries, some are developing COAs, some have developed things that can, they've done the work of trying to figure out what should my endpoint be, and putting their nose to the grindstone to figure that out. And so working with those, I think they've done a really effective job with patient engagement, I think, with engaging their own community to figure things out. And I think some best practices, I think, in addition to just, being sure that you're engaging them early so that their information would be most effective, I think some of the really good work that I've seen are things where there are partnerships between regulators and sponsors and patient engagement groups in the form of consortia. I think CPATH is starting to integrate more patient perspectives into their consortia. I've been working in the xenotransplantation space with trying to understand how patients understand xenotransplantation and clinical trials and what first-in-human trials might look like in that space. And that's been a partnership with patient advocacy groups, with Kidney Health Initiative, the FDA, and the FDA. Kidney Health Initiative is a private partnership with the FDA and the Association, American ASN, American Association for Nephrology. And that has been, I think that is a really good example of how to sort of bring patients into that early stages, those early stages, and start using that information to not just develop, a product, but what is everything else surrounding that going to look like? What could, how are they thinking of the benefits and risk? How are we going to communicate those things to them, to the nephrologist who might be referring them? So that's sort of my advice on best practices. Kind of, work in that preclinical space.
Thank you, Karen and Cara. So this ends our Q&A, and I will give the stage to Larissa for the closing note.
Well, thank you. Thank you, everyone. Thank you, Cecilia and Karen and Cara. Thank you. And good afternoon and greetings to everyone here in the room and to those who are joining us online from various regions of the world. We have come to the conclusion of the first day of the CBER track, and I would like to again express my appreciation to all of the speakers who have participated today, to the CBER moderators, to the conference team, to everyone who supported and made this first day happen. We would like to, so just move the slide here. We would like to remind folks about the one-stop shop of the sbiaevents.com. Please access the stop shop online, and you can get all of the information about this conference from the website here. You will receive the thank you email tomorrow morning, and that's where the activated links will be sent. Please do not forget to complete your evaluation forms for the day. And I also would like to remind folks that the recordings will be available about 10 business days post-event. We have one more day tomorrow that would be filled with various presentations from other parts of the Center for Biologics. They will include topics on vaccine development, blood product regulations, as well as topics of pharmacovigilance and compliance. To hear and learn more about CBER, we invite you to join us tomorrow. And for right now, I would like to wish everyone a great rest of the day, and we will reconvene here in this room tomorrow morning at 8:30 for other CBER presentations, and we will reconnect with our online attendees on the platform tomorrow. Thank you.